Good morning, everyone. Welcome to the 2023 Undergraduate Research Symposium. You'll hear from students today that are part of 3 different programs: the Center for Neurotechnology RU program, the UW Endure program, and the Intel ECE RU program. I want to thank a couple of people before we get started. First of all, our sponsoring organizations. NIH sponsors the UW Endure program. NSF sponsors the CNT RU program, and Intel sponsors the Intel ECE program. The principal investigators for those awards are Dr. Chet Moritz, who's in the front row here. He is the sponsor of our Intel ECE program. Horacio de la Iglesia and I are PIs of the Endure program, and the Center for Neurotechnology sponsors the CNT RU program. This year, the students have been working in labs around the university for the past 10 weeks, doing some fantastic work. We've been discussing it, and they've been creating these presentations for you today. This group was an interesting group this year. I think the best word to describe them was resilient. With the University of Washington financial transformation, they've had to endure some hardships financially because of their awards and things, but they have persevered, and I think you'll be very interested in what they have to say today. The program today is each student will be presenting a 5-minute presentation, and then there'll be 2 minutes of questions from the audience. So feel free to raise your hand, and I'll try to moderate, and you can ask your questions. We also are recording this on Zoom, so there are a few people on Zoom that may have questions as well. So people on Zoom are welcome to ask questions via chat, and I'll relay that to the speakers as well. We are on a very tight schedule, so if your question can't be answered during this oral presentation, between 1 and 3 o'clock, the students will be presenting their work via posters in the room just next door here, as long as they unlock the doors for us. So that will be, the oral presentations will be from about 9 o'clock to noon. Then there'll be a break of about an hour. And then the first set of students will present from 1 to 2. And the second set of students will be presenting from 2 to 3. On the seats that hopefully you're not sitting on them, there's a little piece of paper with a QR code. If you open that up, you can get the program and know who's speaking when, as well as their abstracts, as well as which time period they're going to be presenting their posters, whether it's 1 to 2 or 2 to 3. Okay, our first presentation today is one of our students from the Intel ECE program, Prithvi.
Hello, thank you all for being here today. My name is Prithvi, and today I will be talking about low-cost commercial CMOS cameras for medical imaging. So just a bit of background, the process that the lab was working with throughout the last 8 weeks is called laser spectral contrast imaging, or LSCI, if you will. It is primarily a technique used to analyze blood flow in tissues. And the simple explanation of it is essentially you have a handheld device that will be emitting a laser, and that laser will interact with the rough surfaces of the red blood cells in blood, and the laser light will scatter. Now, those scattering lights will interact with each other, causing what's known as speckles, and these speckles will be captured by an image sensor, typically a camera. And once all those images are captured, they will be ported onto a system that will run various software algorithms based on math that will help highlight the contrast between these speckles. And then you could use that data to assess how blood flow happens. And typically the most useful applications for these are during blood flow changes during disease states or revascularization and other similar states. So the challenge that we were trying to solve with this particular kind of experimental setup is that the cameras that we usually work with or that are usually used in these devices cost anywhere between about $500 to $5,000. Now, for the applications that we would like to use this technology for, that is a little steep. So the goal of the research was to try and achieve the same performances in speed, resolution, and so forth using more cost-effective solutions. So the initial work that was done on the project started out with a Raspberry Pi camera. It is very popular for a lot of computer vision applications, but while testing our custom LSCI algorithms on it, we found that the board would automatically compress any images captured, which is unsuitable for our experiment because since we're working with medical imaging, we would like the images to be in as raw a format as possible because data that is manipulated or compressed or any quality loss could be detrimental in trying to assess how the blood is flowing within the patient. So for that reason, we moved on to a Arduino ESP32 camera. And this time, there was no problem with being able to generate the raw images. However, there was a bit of a constraint and bottleneck with how fast the images were being written onto the SD card. And that was due to constraints with the RAM. So we did some more research and did another thorough inspection of the specs we would need with this new data. And we eventually moved to a OpenMV Cam H7. Now, OpenMV is a relatively new family series of microcontrollers. It's been around for only about 2 years. It is much faster than a lot of the current more consumer grade microcontroller cameras. And it does promise to overcome a lot of the hardware limitations we are facing. And I think this model that we got was purchased for about $85. So it was still within a more affordable price range. So OpenMV has its own infrastructure on how it is used to configure the camera. So we had to rewrite the entire modules for image capturing. We started with just capturing images directly and then automatically writing them to the SD card. However, the performance for this kind of particular method was a little sluggish. We weren't getting the target frames per second rate that we were hoping for. So we eventually moved to another method where we capture images in a stream using a technique called frame buffering, kind of similar to how we take videos. And then once all the images are captured, while certain frames are being captured, previous frames are automatically encoded and stored into the SD card. So it's a lot faster, similar to like how parallel processing works for conventional computing applications. So the method was, I would say, a success. We were able to achieve the speeds that we were hoping for. We were going for around 30 to 40 frames per second. We hit an average about 37, and most common was 35 when we ran multiple tests. And another big advantage with the stream capture method was that it was a lot smoother. And this was really important because sometimes when we were capturing direct images, if the image captured it was not compatible with the amount of RAM in the camera, it would immediately get deleted and moved on to the next frame, which would result in a lot of data loss, which is, again, not very advantageous for medical applications because every small piece of information counts. Precision is extremely important. So overall, I would say the method performed really successfully. The more frames we used, the faster the speed was. We did have some trade-offs, particularly with the image dimensions. Now, like I showed in the previous slide, we did capture them at about 200 by 200, which was sufficient for the experimental setup. However, it does leave very little room for adjustment if we do want to do further tinkering with the algorithms. So it is a little bit of a challenge. And the primary reason for the small image size was we had to sacrifice it with the techniques we used in order to increase the speed at which the camera is capturing the images. And the reason for this was this particular camera's RAM was not big enough to be able to handle both fast capturing with high resolution and high image size. So in order to address this, moving forward, we would like to optimize the results that we got with a slightly more powerful version of the current microcontroller, which is the H7 Plus. We have consulted with the customer support of OpenMV, and they have given assurances that this should be able to handle the same amount of speeds and performance while increasing resolution and image size. So once we are able to verify that, we could move on to designing the hardware portion of the prototype that will handle the laser emitting. It would be done through a PCB. And once both of these components meet the required specs, we can integrate them together and we should have a workable handheld prototype. So thank you all for listening to me today. I would just like to give a special thanks to my mentor, Liban, as well as all the members of Professor Mozeni's EMIT Lab for the guidance, the knowledge, and the support they've given through this entire process. And it's truly been really adventurous, really exciting, and really helpful. Thank you.
One question.
Yeah, great work. Tell me a little more about the application. How fast are the red blood cells moving by? Is the frame rate sufficient to capture the flow rate, that kind of thing?
Well, so okay, just to clarify, application and how, like, the specs of how the blood is moving. Okay, so typical applications, the one we were working with was for burns. So yeah, a lot of the applications for this would typically be for more surface cut wounds in terms of like injuries. Obviously, for one big application for this is usually post-surgery analysis. So after a patient goes through any kind of surgery that requires cutting of any cell or so, we want to make sure that the blood is flowing properly. There's no clots forming that could cause any other health problems. As far as to how fast the blood is moving, it is very varied, but the typical frame rate that we would hope to achieve with this is about 30 to 40 seconds, which is the metric that we were going for. Probably a little more on the higher side is better, obviously, because then we'd have more breathing room to work with. We could do a little more analysis, playing around with the algorithm and so forth.
Thank you very much.
Thank you.
Our next speaker is also an Intel ECE member, and this is Catherine Bennett.
Hello, my name is Catherine Bennett, and this summer I've been working in Dr. Sajjad Moazeni's EMIT lab, like Prithvi, and my project is titled Analog Digital Co-Simulation using Synopsys PrimeSim and Application in 180 nanometer icing machine verification. So to start off, I want to get into the difference between analog and digital circuits. With analog circuits, we think of their signals as having continuous values, whereas with digital circuits, we think of their signals as being binary zeros or ones. These circuits are also designed in very different ways. Analog circuits are designed using schematics like these, whereas digital circuits are designed using code. Both of these are very important, however, since real world signals are really analog in nature, but it's also true that the vast majority of computers and logic are implemented in digital. EMIT Lab works with designs that contain both analog and digital circuit blocks. But most simulators are only built to handle one or the other. Because these circuits are designed in such different ways and they're modeled in such different ways, it's very difficult to simulate them together. And the co-simulator tools which do this are challenging to use. They take lots of different kinds of inputs and control scripts. Good co-simulation, however, allows for more scalable and more complicated mixed signal IC designs. EMIT Lab previously didn't have this capability, so the first part of this project was dedicated to setting that up for them. The first thing we did was we went and we researched different co-simulator tools, and we chose Synopsys PrimeSim as the best fit for EMIT Lab's setup. Then, in order to use this tool efficiently and integrate it into the lab setting, we built a co-simulation environment which takes EMIT Lab's designs and pushes them through Synopsys PrimeSim simulations. And this green flowchart shows each stage of what actually happens when you use the co-simulator tool. Another really important thing we considered when we were making these environments is that analog and digital blocks, their characteristics change as we go through the design process. So for example, on the analog side, we start off with schematics, but as we continue in the design process, we end up with a layout. The layout has much more detailed information about the chip's physical characteristics, like all the different metal layers that end up on there. And so it's a much more detailed and involved simulation. but we need to be able to simulate these designs at any stage of their design process. So what we did was we created 4 different co-simulation environments to enable this. So let's say, for example, if we wanted to simulate our gate level netlist with our schematics, we would use the synthesis co-simulation environment. That's the second from the top. These co-simulation environments will be used in future ImIT mixed signal IC designs. The second part of this project was dedicated to taking this tool and actually using it on a 180 nanometer chip. This is the icing machine, and its block diagram is on the top left there. As you can see from the block diagram, this is a very mixed signal chip. It has both digital and mixed signal blocks. The icing machine, the purpose of the icing machine is to optimize the max cut problem. With the max cut problem, you have an input graph of nodes and edges, and the goal is to find a path through that graph which cuts through the maximum number of edges. The icing machine does this through something called SRAM compute in memory, and it does it through recursive computation. This chip is currently non-functional in fabricated hardware, which has created a need for co-simulation. With co-simulation, we can perform an in-depth characterization of the design and all of its internal workings, and we can start to gather some data on how well it actually solves these problems. We took a progressive approach to testing, where we started off by testing subblocks and gradually moved up to testing the full chip's operation. Here are some example results from when we got to that point of testing the full chip operation. You can see in the top left, the 60 node graph that we were testing on. And right below that, you can see an example of the icing machine actually optimizing for that problem. And you can see the max cut value increasing as it goes through its iterative computations. The pink graph is, it illustrates how well and how often the icing machine found good solutions across various tuning parameters, where darker regions are regions where our tuning regions where the icing machine performed better, and lighter regions are tuning parameters where the icing machine performed worse. But throughout this process, we verified that the icing machine's design is actually functional, and we started to get a good idea of how well it performs. And future work will involve continuing to investigate performance and also further investigating why the hardware is not behaving as the design does in simulation. Yeah. Thank you, and thanks to ImIT and the Intel for sponsoring this, and the ECE department for facilitating. Any questions?
Questions for Catherine?
Just curious a little bit on the algorithm to like have the line go through the various nodes. Could you maybe elaborate on like why that's needed?
Like what the application of the icing machine and is? Yeah, so the max cut problem, it's a pretty general model, but it's very applicable to, there's a lot of applications in finance and in economics and in management of very large and complicated systems. It's very convenient that, it just happens to be convenient that it fits a lot of different applications. Yeah.
Thank you, Catherine. Our next presenter is Caden Borland.
Hello, everyone. My name is Caden, and this summer, my lab and I studied an approach to spatially variant deconvolution for real-time image processing. First, let's talk about what deconvolution is. Deconvolution is the inverse of convolution, a mathematical way of combining two signals into one. All imaging systems have this convolution inherent in it, convolving the ground truth with something called the point spread function, or the PSF. Then when the ground truth and the PSF are convolved, each pixel is now an average of the ones around it, and this is weighted by the point spread function. Even because of this, the final image comes out blurry, And even the best cameras have this blur just on small scales. So then the question is, where does this point spread function and where does it come from? Well, it comes from the physical limitations of imaging systems, as well as the physical properties of light. Single point sources cannot be represented as points, but must be represented as spread functions. And the question is, how do we sharpen this image using the convolved image and data on the point spread function? We already have current methods for this, and examples of that include the Richardson-Lucy, the Weiner filter, and many others. I even tried to create a custom algorithm myself this summer. And after studying their effectiveness for quality and for runtime, I found that the Weiner filter was the best suited for this application. And that's because it met the criteria of a runtime of under 0.1 seconds, which was important for real-time imaging. Then on to the real problem, spatially variant deconvolution. In recent developments, new lenses have been made that produce a significant spatial variance in their PSFs. Then what does this mean? Essentially, the deconvolution is the same process as before, but the PSF changes based on where in the image you're at. If you're in the middle, it might look something like the center, more circular. And if you're near the corner, it might stretch out. And this property is what we call spatial variance. Then when you convolve the ground truth with the spatially variant PSF, it produces an image that is blurry, not only blurry, but blurry in different ways in different parts of the image. If you'll notice in this image that the border is slightly more blurry than the center because of this convolution process. So then the new question is, given the data that we have data on the PSF, how can we take the convolved image and the data to create something that is sharper and more clear? This question was actually the bulk of my research this summer. And this is the method that my lab and I came up with. First, we would section off the image into a grid. And this grid could be 3 by 3, 5 by 5, 10 by 10. It depended on the image for what was best. Then after we had those sections, we would expand those sections for 2 reasons. One reason was that the Weiner filter would have enough data to be accurate. And the second would be to reduce the border noise that's inherent in the Weiner filter. Then after we have all of these sections, we would perform a local deconvolution on it using the corresponding PSF. In this example, I have the top left corner of the previous image and the top left corner of the PSF. And after we perform a Weiner filter restoration on it, we can then take out those edges off and collect each deconvolved segment and stitch them all together to produce a final image. Then what we're left with is an image that is sharper and clearer than before. If you see in the second image, you can see a bit more detail than the first. And using various metrics of image quality, we found that this method can produce a 2% increase in the quality of the image. And while that might not sound like much or a lot, it proves that this method is a valid approach to tackling the problem of spatial variance in deconvolution. And that further resources and time could be invested into it to make it better and more effective. Thank you for your time.
Context of image processing, what potential applications could you see this being used for, and maybe just how this convolution and deconvolution?
So for this lab specifically, there is, like I said, there is a new lens that is being developed that produces significant spatial variance. And we don't currently have many methods for tackling how to deconvolve to make images sharper. Deconvolution is already pretty widespread to make images better, but for the problem of the spatial variance, we needed a new method. And I know that this lens is going to be used in an endoscopy camera. And because the lens is actually 2-dimensional instead of curved, which makes it more lightweight and smaller, which is beneficial for medical purposes. So yeah, we needed a way to post-process those images. And because there's such little data that we're working with, which is I think 500 by 500 pixels, we need like all the information we can get from them. So even a slight increase in quality is effective.
Thank you, Kate. Our next presenter is Emma Tran from Josh Smith's lab.
Hi, my name is Emma Tran, and this summer I worked on camera localization on acoustically levitated particles. So first, for my introduction, a little introduction into acoustic levitation, something my mentor has been working on. It's pretty much suspending small particles with ultrasonic standing waves. Overall, it could suspend something less than 2 millimeters in diameter, and overall it effectively neutralizes the effect of gravity on the object, so it levitates. And what I worked on was the visual localization of said small levitated particle. Here are some pictures taken of different objects it could levitate, such as like leaves, insects, and microchips. Next, I'm going to dig into the hardware I used. So here's something that was set up this summer. It was the bulk of my research. So first, the camera mount that my mentor 3D printed, we attached 2 CSI cameras, as you can see kind of farther back to it. They're Arducam CSI cameras, as well as a Jetson Nano we attached at the top, which pretty much acts as a computer, and the acoustic levitation rings, which are in front of the camera, so it could capture that. And more about the Jetson Nanos. To the right, you can see the Jetson Nano previously in the last picture, but to the left, as the summer went on, we need to move to the Jetson Orin AGX just due to the increased memory and RAM power, since the Jetson Nano was actually overheating and it couldn't process as many cameras as we wanted, because we ultimately needed at least 3, ultimately 4 cameras to do the things that we needed. Then for my camera display, this is just an image that I took of the detection system that I used OpenCV and Python to create. It provides real-time tracking on the object with accurate visual insights, and could be used on particle manipulation. So as you can see in the picture here, it just pretty much gets the center point of the object as well as outlining where it is detecting. And I just put a small piece of paper in there just to test it and to take a picture of. Then why would we need this? Its purpose, overall, object localization. We can get things like visual feature extraction, such as color and shapes of the objects, as well as overall a platform for more robust controllers to develop a feedback loop for acoustic levitation controllers. Overall, the many transducers, we want to get a loop for them, since the acoustic levitation device consists of so many of them, and overall end up with visual features extractions and the coordinates and the localization of the object. So what's next? Next steps for this project would look like. So we would need better cameras. We're thinking about pinhole cameras due to the ability to create sharper images to better capture small particles, and as well as better using OpenCV static image subtraction as well to isolate those levitated particles so we can detect them, so it's easier since they are so small, as well as camera calibration. Charuku board camera calibration actually is something we've been looking into. It's a little bit better than the standard checkerboard calibration, just so we can get better, more accurate points, as well as depth instead of just X and Y points, which is also what the 4th camera would be needed for. So here's just an image going into the future. The 4 camera setup, we would ultimately, these are just plain USB cameras, which is something else that I worked with this summer, but they weren't able to do what we needed them to do. So ultimately what we would want is for pinhole cameras, since they have a smaller lens, and as well as this is the camera mount that was printed. It would pretty much go atop the device, and the acoustic levitation device would go inside, so we can track those particles. And as I said previously, there are 4 cameras, so we can get not just the X and Y coordinates, but also the Z depth and have that be accurate, as well as a computer designed around USB limitations. Since this summer, a very big problem was running into limitations with the USB. Since they are USB cameras, they go into one USB port, and most computers have one USB hub, and it can't really process more than roughly 2 of these cameras is what I ran into a lot this summer. Due to time, and for my acknowledgements, I just like to thank the lab, Joshua Smith's lab, my mentor Jared, and Intel for funding this project. Thank you.
Questions for Emma?
Go on, Eric.
Hi.
Fantastic work. I'm fascinated by the fact you can levitate things with sound waves. Is the current system, is it open loop? Is it just blast it with sound waves and hope it floats around? And is your camera then needed to figure out where the particle is for closed loop?
Yeah,
Or is there some
pretty-
other way to sense the location of the object?
Yeah, pretty much what I've seen my mentor Jared do. He knows a little bit more about this than me, but pretty much it levitates a small particle, and he works on different ways of doing so. So what the camera would be used for was like, let's say like something we test on frequently is like water droplets. Like if it splits or something, we want to detect that, and maybe later into the future, like color, shape, or like maybe even object detection. Like, is this a bug? Like the insects that we were talking about, that as well.
Time for one more question.
I have a question.
I think it's
Oh, go ahead.
Hi, this is Josh. Good presentation. Thank you. Wondering, you were talking about the limitations of getting a couple of multiple cameras into a computer. Do you think you could connect up multiple computers, or is there too much communication required between the computers to make that work?
So yeah, what we were planning on doing, or what we were recommended on doing, is maybe building a camera with like, so the USBs would go to different hubs. So maybe possibly like 2 different computers wouldn't really be necessary, I guess. But we were thinking about putting different processors or hubs into the computer, so they wouldn't have the same USB hub internally, so it wouldn't run into that limitation. But we're still looking a little into it. But that's what we were planning on trying next.
Thank you.
Thank you.
Thank you.
Our next presenter is Myra.
Hello, my name is Myra, and this summer I worked at the SQR lab with my mentor, Sarah Meridian, characterizing the Gaussian beam. So first, I'd like to introduce the ion trap. The ion trap is one of the methods used for scalable quantum computing, where we trap the ion inside of a vacuum chamber using an electric field. And in this electric field, where the ions are staying, we then manipulate the ions electronic state in order to represent any qubits. So qubits are similar to like regular bits in a computer, but in this case, it's a quantum computer. And we manipulate the electronic state using 6 different lasers. So as you can see in the image over there, there's a photoionization laser, which is the 375 and the 422. Then we have the cooling beam, which is the 397. And then we have the qubit transition laser. This is the one that simulates the qubit transition from like 0 to 1. That's the 729. And then on the top, we have the repump and reset laser, which are the 866 and 854. And what I worked on this summer was being able to create a setup outside of the chamber where the objects are set up and focusing onto the ions inside the chamber. And they're focused inside the chamber and as well as onto the ions. So in order to do this, I had to understand how the laser propagates over time. And all the lasers that we have, and majority of the lasers propagate in the form of a Gaussian beam. The Gaussian beam just means it has this shape where it has a minimum point of focus and then expands back out. And this minimum point of focus has the highest intensity. And we want this point of highest intensity to be focused on our ions in order to be able to create those electronic transitions. And this minimum waste is, we want to be able to locate it where in the position it is, because the laser has to travel a certain distance and to be focused onto the ions in the center, as well as being able to have it to be under 100 microns. 100 microns, to put it in perspective, is the thickness of a hair is 70 microns. So we have to make sure that the lasers are all focused at the same point inside of the chamber at that small distance or diameter. So the optical setup that I created is the one that you can see on the side where it has multiple beam splitters and mirrors and different lenses that are all used to be able to focus the laser beam onto the ions in a specific location. So with different manipulation of the lenses, we're able to move the waste wherever we want of the ions, as well as with the different mirrors, we are able to get different degrees of freedom, where we're also able to change the distance of the beams in the X and Y direction. If we're able to, so we're able to manipulate the location of the focus of the beams onto the ions to ensure that they're all co-propagating. And with this, we're also able to optimize and ensure that all the ions are being hit by all the lasers, and ensuring that the minimum waste is focused onto the ions. And in order to do this, we take multiple measurements where we have multiple images that look like this at various different distances after the window. And then we do a beam profiler where we're able to measure the waste, which is, the waste is just the diameter. So we take multiple images at different distances after the window. We do the beam profile, and then we're able to characterize this. And as you can see in the image all the way to the far right, is we're able to identify the beam waste as well as where in its propagation state it is, to be able to identify like, oh, okay, where are the ions inside the chamber, and being able to ensure that, oh yeah, the waste is going to be focused at that point. So I was able to characterize for the optical setup, the 3 different lasers, and achieve them to be under 100 microns. And they're all 1 millimeter within each other. And then some future work would be being able to adjust the optics outside of the chamber once the chamber is ready, since the chamber currently is not, as well as doing the physical setup for the other 3 lasers. And then finally, a potential future project could be fabricating an attachment onto the chamber to improve mechanical stability than just having the optics outside of the chamber. Thank you. Questions?
Questions for Myra? In the back?
Really nice work, Myra. The big question for quantum computing is always scaling, right? So do you feel like your approach may scale to lots of qubits, or is there a limitation in terms of how many qubits you can potentially manipulate and change the state?
Okay, I'm not too familiar with that, since I just worked on focusing the lasers onto the ions, so I wouldn't be able to fully answer that. But we could talk about it later, as I could also ask my mentor. Yeah. Thank you.
Thank you, Mike. Our next presenter is Maddie Hernandez, who's one of our UW Endure students.
Welcome everyone. My name is Maddie, and my experiment and presentation today is over stress and how that induces a signal in calcium and dynorphine in the colostrum. So in a 2022 survey by the American Psychological Association, with a total survey population of 2,051 American adults over the age of 18, 87% of that total population said that their major causation of stress was due to economic fluctuation. 81% of that total population said it was due to global uncertainty, as well as geopolitical strife and COVID-19. So we can see stress induces a change in our sensory perception. Everything that we input has a direct correlation to our output. So an interaction with something pleasant can change based on the environment. And that's really what my experiment was working on. So looking at dynorphine is responsible for pain, addiction, and mood regulation, which is stress processing related. And dynorphine is a specific type of neuropeptide that interacts with GPCRs. And GPCRs are G protein coupled receptors. And they're 7 membrane-based protein receptors that mediate downstream signaling for hormones as well as neurotransmitters. As we can see here, we see the word core. What is core? Core is the kappa opioid receptor. And the kappa opioid receptor is a specific receptor for dynorphine. So it's able to regulate the amount of dynorphine that's being released and binded with the pre to postsynaptic cleft. The technique that I utilized for my experiment was what's called fiber photometry. So fiber photometry is underneath the umbrella term of optogenetics, and that's how we stimulate the brain using light. So here we have an imaging probe, and the imaging probe is implanted into our population of mice. And then we have a fiber cable that's connected to our imaging probe here, as you can see. And light is transmitted through this cable into the imaging probe of the brain of the mouse. And then we're able to see the fluorescent signal within the brain. Okay, so the experimental setup, we're specifically looking at the claustrum, and the claustrum connects the cerebral cortex to the amygdala, the thalamus, as well as the hippocampus. So why are we looking at the claustrum? Core, or what I said before, the kappa opioid receptor is highly expressed in this brain region. And how we're able to look at non-specific neural activity is through what we call GCamp. So for this experiment, we're looking at stress in non-stress induced environments. And in order to induce the stress environment, we have here is what we call the forced swim experiment. So each mouse is individually placed in a bucket for 15 minutes and then a 30-minute rest period. So once we have our stress and non-stress induced environment, we have two types of sensory cues. We have an attractive sensory cue, which is the female urine sensory cue, along with the tail lift, which is the aversive sensory cue. So looking at GCamp is non-specific to neural activity. And here we can see that from these 2 points, that there's elevated neural activity in the claustrum towards tail lift under stress conditions compared to non-stressed conditions. And we can see that it has a similar response as well to the female urine and how it is an increase in stress-induced response compared to the non-stressed. Okay, now looking at K-light, what exactly is K-light? So K-light is a indicator for dynorphine, or it's a way that measures dynorphine release. So here we see that there is not as much of an increase, as dramatic of an increase as we see in GCamp. And this can be due to a multitude of hypotheses, but one of the hypotheses that my mentor and I came up with was that in this highly induced stress environment, we have an increase in dynorphine, right? So because of this increase in dynorphine, it fluctuates our baseline level, or the regulated amount of dynorphine that's released in the brain. So because of this fluctuation in the baseline, it tampers with our results of stress and non-stress induced environment. However, we can see here from the female urine response that there is a positive correlation between the stress compared to the non-stress induced environment. So something that feels pleasurable to us still feels pleasurable even in highly stressful environments. So in conclusion, we're observing how GCamp and K-light responds to tail lift as well as female urine sensory cues in stress and non-stress induced environments. And our experiment further extends the question of examining the effects of stress specifically in the claustrum and how we can test our understanding of stress and how this can impact neurogenetic diseases, brain function, how our brain interacts with drugs, and the list continues. My acknowledgements are to the Endure program, the National Institute of Health, along with making it possible by the Brucus Lab with direct mentorship of Jinky Chen. Thank you.
For Maddie.
Yeah.
I have a question about your experimental design.
Yeah, sure.
Why did you choose to use the female?
As an attractive sensory cue?
Yeah.
Well, because
about mice and things.
yeah, no, of course. So even with the male and female species of mice that we tested, so both genders, both have the same kind of attractive sensory cue. And mice have a different type of olfactory respond. So they have a more enhanced sense of smell, as that's like their main basis of sense compared to ours. It's like eyesight. Their eyesight's not very good. So both the tail lift and the female have different or opposing types of sensory cues. So we can see from both genders that it has a positive correlation of dynorphine. Yes.
And I just have one question for you. The claustrum's not really understood, and there hasn't been a lot of work on claustrum.
Yeah,
So can you give us a little bit more information about what you think the claustrum might be doing? And what's its function, I guess?
Yeah, right. So as I said before, so we see the kappa opioid receptor being highly expressed in this region. So we hypothesize that there's some kind of correlation with stress and how that has to do with stress. But yeah, there's not a lot of scientific literature out there about the claustrum. So that's what we hope to further extend our research in this brain region. Yeah.
Thank you, ma'am.
Thank you.
We'll end this first session and it'll be with two more presentations and there'll be a short 5-minute break. And the next two presentations are from the De La Iglesia lab.
Hi guys, thank you for coming. I am Vivian Chen. I'm from the De La Iglesia lab, and I'm investigating whether amygdala optogenetic stimulation induces changes in behavior in CFOS expression. But before we get into that, I thought that I'd give you guys some context into the background that our lab specializes in, which is circadian rhythms. So in circadian rhythms, we have these things called zeitgebers. The most well-known zeitgeber is the light-dark cycle, and those entrain our biological oscillators, so our circadian rhythms, and that manifests itself in our rhythmic outputs. So when we sleep and when we're active. It's important to note that while the most well-known zeitgeber is the light-dark cycle here, which is done by the sun, there are other non-light related zeitgebers out there too, such as fear, which is what our lab recently discovered. And to do that, we use a novel fear chamber. In this chamber, we have a safe nesting area where no fear stimulus is being delivered. And then we have a foraging area where the mice are active, where fear stimulus is being delivered. And how this behavior appears under this paradigm is through these actograms. So in this actogram, we have rows that represent 24-hour periods, day periods. And it's been double plotted so that we can compare the light and dark cycles a lot easier. So the light cycle is indicated by this beige color here, and the dark cycle is indicated by this gray color here. Periods of activity are indicated by these black bars. So as we can see here, this animal is relatively nocturnal. When we deliver fear stimulus during the day, indicated by this purple, this animal becomes very, very nocturnal with almost no activity during the light cycle. But when we deliver fear during the dark cycle, when this animal is normally very active, we can see a shift in their behavior. They become actually very active during the light cycle. And what's even more interesting is when we remove all of this stimulus and we let their circadian rhythm free run, meaning we have no stimulus, including light, and we let their circadian rhythms to just free run in the darkness, this pattern of activity persists. So that means that which is indicated by these bouts of activity here. So that means that this animal circadian rhythm has been entrained by the fear stimulus. So that means that there's some kind of fear-related oscillator somewhere in the brain, and we are really interested in understanding how that works mechanistically and where that's located. And we thought the amygdala would be like a very reasonable place to start because it is a fear processing region of the brain and the fear circuitry. So to do that, we used optogenetics. And what is optogenetics? Optogenetics is just virally expressing channel rhodopsin, which is a light sensitive channel protein in the basolateral amygdala, the BLA of the mice. And then we have this LED wireless probe that uses light to locally activate these neurons in the basolateral amygdala. And here we have the same setup, the same paradigm, but instead of using foot shocks as a fear stimulator, we are directly stimulating the amygdala using optogenetics. And we are hoping to see the same kind of behaviors that we saw here with the entrainment patterns. But because we didn't really see those observations and behavior, we thought that maybe we need to make sure that the amygdala is actually being stimulated. So we decided to look at neural activity, which is where I did the bulk of my work, as you can see here. So immunohistochemistries allow us to see CFOS expression. We use coronal sections of the brain and we stain them using primary and secondary proteins that are specific to CFOS. And CFOS is just a marker for neural activity. And then we image those coronal sections using an A1R confocal. And those results are seen here. So we have the left amygdala and the right amygdala. The left amygdala was the only amygdala in this section being stimulated, which is confirmed by the presence of M-cherry. And as we can see here, there's a general trend that when stimulation occurs, when M-cherry is present, there is a higher expression of CFOS. So there's more neural activity when that region is being stimulated. However, because we didn't really see any robust responses and behaviors, we didn't see entrainment occurring. We are looking at stimulating other parts of the brain region and hoping to see the same entrainment responses that we saw with the foot shocks. And then from there, we can find where the fear entrainment oscillator is located. So yeah, and none of this would have been possible without these people. So a huge thank you to them, and I'll take any questions.
Or Vivian? Yeah, Brian?
Yeah, so essentially you confirmed that the amygdala, which typically is associated with like fear, is not responsible necessarily for these behaviors that you saw, the fear shock. So what other areas might you hypothesize or test for in future with optogenetics to see if they're involved?
We are currently looking at the periodontal gray area, which is also part of the fear circuit of the brain. And that's what we're doing right now. But yeah, that's really what we're looking at. There are other parts of the fear circuit, but we are just like looking at that one right now.
One quick one, go ahead.
You said that fear is like an oscillator for circadian rhythm, I believe. Like, when you remove that fear, and you said it persists, do you know how long and if it disappears eventually?
So as we can see here, this entrainment, each row is a day, and we saw that this fear persists throughout the rest of when we were recording this. So I think it can be assumed that this activity sort of persists throughout the rest of this experiment while these animals are in this fear chamber setup.
Thank you. And our last presentation before a quick break is from Alec.
Hello, everybody. This summer, I worked on a machine learning approach towards seizure prediction in epilepsy, in a mouse model of epilepsy. So the specific type of epilepsy we were looking at is Dravet syndrome. Dravet syndrome is among the most severe types of epilepsy and is caused by a mutation in the SCN1A gene. And so it is characterized by early life seizures and resistance to many common treatments for epilepsy. So the goal of our project is to create a closed loop seizure prediction device that can predict the seizure and then prevent it for its onset. In this diagram here, you can see that we would gather the neural data from the mouse and then run it through our machine learning Python model. And then we would have a stimulus for the seizure intervention to hopefully ameliorate some of the issues that come with long-term seizure damage. And so for some background, there are 4 different stages of a seizure. There's the interictal, preictal, ictal, and postictal. Ictal here means seizure. And so the interictal is the baseline. This is where the mouse shows no sign of seizure, and that's where the mouse typically is. Then the preictal phase is the phase right before the seizure. The ictal phase is where the seizure actually happens, as you can see in this increase in activity where the neurons are firing rapidly. And then the post-ictal phase is characterized by this lessened activity after a seizure. What can happen sometimes is a mouse will go in between the interictal and preictal and then go back to interictal without actually having the seizure. And so the way we approach this machine learning problem is we decided to go for a supervised learning approach. We would label 5, 10, and 20-minute increments before the seizure as pre-ictal. Then we would label the ictal and post-ictal, and then every other value would be interictal. And so from that neural activity I just showed you, would then extract 98 time and frequency-based features, for example, power, amplitude, and voltage, to then train 10 different machine learning models. And so the way machine learning works for our purposes is the models are trained off of these 98 features and then find the best quantitative relationships between the features and the labels to then generate a prediction of a seizure stage. And so after we had tested all of the models, and when you test a machine learning model, what you do is you apply the predictions to a data set that it has never seen before. So it is essentially creating predictions, and this would get a accurate score of how it would perform in real time. And so what we found is that this light purple box here, this is a box and whisker plot. This is the LDA-SVD, linear discriminant analysis. We found that the test accuracy was about 64.5%. And it also had among the highest prediction times, about 0.0001 seconds, because it's on a logarithmic scale. And this is really important for our project as we want to eventually have this be real time. So fast prediction time is necessary. This figure on the left here is a confusion matrix. It shows the algorithm's predictions versus the labels I had labeled. So if you focus on this colored boxes in these diagonal, and then on the right here, I labeled 15,794 values as interictal. And on the diagonal, we find this thing called recall, where the algorithm and I agreed on the label. And so 5,653 out of the 15,794 labels, they labeled it as interictal. However, it also classified some of them as wild type and preictal, which makes sense because wild type is a mouse that has no genetic mutation for Dravet syndrome. And so it should misclassify some of them as interictal or wild type because there should be overlap between the interictal stage and the wild type mouse. And then also, it makes sense that some of them were classified as preictal due to the fact that I only labeled the 5, 10, and 20 increments before a seizure as preictal. And so if there was no seizure, I would not label it as preictal. But as we get closer and closer to the ictal phase, we can see that the recall becomes much higher, with about 70% in preictal and then 80+% in the ictal and postictal. And what this shows is this could be a promising method for seizure prediction. And this graph right here is when we applied the predictions to an actual seizure. So this focus on this line called label data. The blue here is interictal. The yellow would be the 20 minutes before a seizure. Orange would be 10 minutes before the seizure. Light red, 5 minutes before the seizure. The dark red would be ictal, and then purple would be postictal. And then if you look at this line that says 5-minute prediction, as you get closer and closer to the seizure, you can see that the light red becomes more and more predominant, showing that the model generally understands what the temporal progression of a seizure is. And so for future, we need to test different algorithms and potentially use neural networks, which have been shown to have a higher classification rate for seizure data. And we also need to gather more data on different mice and more seizures to make the algorithms more robust. I want to say thank you to Dr. Chudler, Dr. De La Iglesia, and Assad Beck for their help on this project. And yeah.
For Alec.
You mentioned the need for fast model response times.
Mhm.
How quickly does the model actually need to respond in order to successfully intervene?
So what we've been doing is we've been gathering, we extracted features in 5-second intervals. And so the problem is that extracting these features takes some time. So if we were extracting 5 seconds for features, then we would need to have the prediction time be very fast, because extracting the features can take a very long time. It's actually something we're working on right now, because it requires fast processing power to get the EEG data directly and then extract the features.
One more question, Josh.
Nice work, Alex.
Thank you.
One of the big issues with this kind of classification is can it transfer to, for example, another subject in your
Mhm.
another mouse?
Yeah.
The data you showed, was that for a single mouse or was it across multiple?
So for the data, we actually used 4 different mice, and they were all recorded over the course of 30 days. So there was a previous study with Dravet syndrome where they had gathered data for 4 different mice with Dravet syndrome. And so in total, we used about 20 seizures worth from the mice as data. And then we also applied the wild type data so that the model would become more robust and could classify things at a higher rate. So we do need more data because it's there weren't as many seizures as there could have been, but with the test accuracy being about 64%, we're hoping that this could lead to a better like real-time prediction model.
Thank you, Al.
Yep.
Okay, this is our first short break. We only have 5 minutes, so don't go too far. If you are leaving, remember that we have posters from 1 o'clock to 3 o'clock. So please come back for the next talk, which is in 5 minutes at 5 minutes after 10. So don't go too far. Are you up next? No, yeah. Yeah, everything's been updated.
Okay, so before I'm a little bit nervous. Is this one, right?
That's forward, and that is your laser pointer.
Okay, that's the laser pointer. Okay, and this is the
But
pointer.
we can start yours.
Yeah.
Just go backwards.
That's great stuff. Maddie, right?
So glad to go.
Yeah, you did a great job.
Oh, very confident. Mistakes off right. Oh, 80%. What?
I think I have a timing. It's not, they are not seeing anything? Okay.
All right, if everyone can find their seats again, if everyone can find their seats again, we'll get started. Thank you. Our next speaker is Neela Carey, also one of our UW Endure students.
Thank you. My name is Neela Carey and I'm a computer science student at UW. During the last 10 weeks of my research in PEMP lab, I was working in investigating the feasibility of utilizing fiber photometry during the cocaine self-administration. And the reason we focus on cocaine is because the national overdose deaths has been increased in last 2 decades. And there are inadequate treatment for cocaine. We are studying neurotransmitter here to understand the neurobiology of addiction and find treatment for that, and in our case is for addiction. There are 2 techniques to measure the neurotransmitter. The old technique that has been used in our lab and in other labs is a voltammetry, which is the electrochemical technique which measures the change in a current during the redox reaction of the dopamine. In this technique, we can monitor the neurotransmitter release in real time, and it can provide insight into dynamic chemical change in a specific region of the brain. The old technique, in the old technique, we cannot measure all neurotransmitter. That's why we are using the fiber photometry, which is the optical technique that measures the change in a fluorescence. We can monitor the neural activity during the genetically encoded protein. A little explanation, how is this technique work? So normally we only have a dopamine receptor in a postsynaptic area. But since we have a D-light receptor and we have modified our receptor, we are exciting the fluorophore and we can record the signal through the optic fiber. For this, we designed an experiment to find out about this neurotransmission. So we had male rats. We did a bilateral viral injection and optic implant in the nucleus accumbens, and we used the cocaine self-administration in order to see the dynamic of the neurotransmitter. Actually, no spoke here is the no spoke that animal received the cocaine voluntarily, and during that time, he received the light and white noise. The timeline for this experiment, so we did a surgery, we had to train our animal. Animal had to have 10 consecutive no spoke during 3 consecutive days to start the experiment, and we had a short access and long access, and we measure our data. So the old technique, so in order to validate our data, we did that experiment, and as you can see here, that the dopamine is increasing in the fiber photometry, which is aligned with the previous literature has been done in our lab. Also, for the long-term access, we had a very similar data that are dopamine is increasing right after the no spoke and a fiber photometry, which is very similar to the old technique in the field of voltammetry. In conclusion, we use the D-light and we use our primary data to see if it's matched with the previous literature in the field of voltammetry in a short-term and long-term access during the 3 weeks of the cocaine self-administration. For the future direction, we can utilize fiber photometry in a conjunction with the other modified receptors such as D-light for the detection of a different neurotransmitter and enabling the dual recording involving other genetically modified receptor within a single brain of region. And these are the capabilities that voltammetry lack. And thank you so much to the indoor program and PEMP lab that helped me and support me during this research. Thank you for listening. That's it.
Questions for Neela? Ryan?
So it seems like you essentially validated the method compared to the previous, which is voltammetry, right? I'm curious if there's anything from the new method that you saw that was, I guess, different or more insightful as to like responsive for cocaine, any like advantages to the method?
So there are some, but there are some different, but we cannot generalize that because we need to find more data. These are just the primary data and we cannot go further than that. Mm-hmm.
Thank you, Neil. Our next presenter is Sarah Shader. She's one of our C&T RU students.
Hello, I'm Sarah Shader, and I will be presenting on my project from the summer, an interface for evoked potential experiments with the cortex-brain interchange. So first, what is an evoked potential? It is an electrical signal recorded from a specific area of the nervous system in response to a stimulus. And so there are different types of evoked potentials defined by different types of stimulus and where the electrical signal is recorded. But most relevant to my project are cortical evoked potentials. And so CCEPs measure activity in one cortical zone in response to stimulation in another cortical zone. And on the right there, I have a picture of some examples of CCEPs so you can get a sense of what that electrical signal can look like. And one of the reasons we care about CCEPs is they have the potential to characterize connectivity between different areas of the brain. And so my lab is currently working on a treatment for stroke rehabilitation. And since evoked potentials have the potential to characterize connectivity, we hypothesize that we might be able to track the progress of treatment by seeing how connectivity is changing and hopefully improving throughout the treatment. And so that is what motivated this project, was the need for a way to administer those evoked potential experiments to hopefully track progress. And so my application is designed to work with the cortex-brain interchange. This device is an upcoming investigational device, and it sends stimulation and reads activity. And the brain interchange has high temporal processing abilities, which is important when you're working with evoked potentials, which need high precision in the time domain. And so the brain interchange is made up of a cortical grid, which contains the electrodes that are sending stimulation and reading the neural activity, and that is fully implanted. And then there's an electronics unit and an external unit, and lastly, a computer, which is where a user would be interacting with my interface for administering evoked potentials. And so I designed a Windows application to allow for straightforward clinical monitoring of evoked potentials using the cortex-brain interchange. So on the left, we have controls to define the experiment and display parameters. At the bottom, we have a console which updates the user on the state of the experiment. The chart at the top displays the data that's streamed from the selected electrode channels, and the chart at the bottom is a real-time average. And so what that chart is doing is taking a sort of snapshot of the data every time a stimulation pulse occurs and adding that into a running average. So as the experiment runs, every time a new pulse is sent, it's factored into this running average. So we are getting this constant real-time update. And the reason we care about an average with evoked potentials is as we have more and more data, as we average it out, we cancel out some of the noise and are left with a more robust electrophysiological response. And so here I have a demo of my application. And so this was recorded just on a test system that's open to air and not using actual electrophysiological data. So we won't see any evoked potentials in this video, but you will see the spikes from when the system is applying stimulation. So you can select the source and destination electrodes for bipolar stimulation. And then we can toggle which channels we want to display data from. So you can select multiple channels at once and deselect and select at any time. And then when we start stimulation, you can see the activity being streamed at the top, and at the bottom, we have that constantly updating real-time average of the signal. And so in addition to the application, I built an offline pipeline in Python to perform further signal processing to get evoked potential results. And so while the experiment runs, data is logged, and then we can take that data and put it through this pipeline. So the pipeline applies some gentle filtering. It normalizes the Y scale and does a baseline correction to center the signal at 0, and then averages every trial under the same condition to get one final evoked potential result. And the goal of this pipeline is to reduce noise and isolate a more robust response. And thank you all so much for listening. And special thank you to Dr. Treadler and the CNTRU, and to Hanbin and Jeff for all their support and mentorship this summer.
Questions for Sarah? Raj?
Yeah, nice work, Sarah. So one of the questions here is, when you actually port this to an actual patient setup, there may be other challenges. So do you foresee translation of what you've done here and shown in simulation versus what are the challenges you expect when you actually now start to the patient?
Yeah, it's a great question. I'm not sure, honestly. I feel like the process is so early, like it hasn't even been tested on like an animal or anything, but I guess, yeah, honestly, I don't know, but it's a great question. I'll think more about it. Anyone
And what
else?
about going beyond just one electrode to another electrode? Like typically on a screen for multiple sites, right? So
Yeah, so I have, right now there's 2 modes that you can implement. And it's one of the controls on the left that you can toggle between a scanning mode and then a single experiment mode. So when you're in the scanning mode, you load in a configuration file with a sequence of different channels that you want to test, and it'll just cycle through all of them, and then you can offline analyze that data. And then you can also be just in a single experiment mode where you're like manually selecting which channels you want to stimulate.
I have a question for you. It looked like in your setup, you're stimulating quite close to your recording site. And it looks like you're having quite a bit of artifact in at least the first 200 or so milliseconds. Are you trying to eliminate that artifact anyway, or is that some of that filtering is for?
Definitely. So right now, like the demo I filmed, I don't think if you were actually conducting an experiment, you would be recording from those channels. I just did that so you could kind of have some sort of data to look at when you're seeing the averaging. But yeah, in general, you would want to be looking at not necessarily the source destination and the source channel, and there would be in the filtering process like an artifact removal.
Okay, thank you, Sarah. Our next presenter from Similar Lab is Arsh.
Good morning, everyone. My name is Arsh Banapal, and this summer I had the pleasure of working in the GRID lab, specifically under Dr. Jeffrey Heron, and looking at how emergence, which is the regaining of consciousness from anesthesia, works, and how we can use this information to help train a machine learning classifier that can predict the stage of consciousness from emergence data. With this being said, the first question we want to answer is how does anesthesia, and in our case propofol, work to induce unconsciousness? So propofol works in 3 main ways. The first is it slows the glucose and oxygen consumption in the thalamocortical and frontoparietal networks. Second is it decreases overall brain connectivity. And the third is it reduces the cerebral blood flow. As you would expect, during consciousness, we would essentially see the opposite of these effects. And the main thing we want to know with consciousness, in this study at least, is where does it reside? So consciousness is mainly attributed to 2 brain structures, the brainstem and the thalamus. The brainstem is important for many vital functions of life, especially consciousness, and the thalamus is vital for processing information from our body senses. And these 2 brain structures are located in the thalamocortical network, which, as I described before, is where we see a large decrease in activity due to propofol. So this is very important for our study. So the data was collected from a pediatric patient who had epilepsy, and they had 12 electrodes implanted in various regions of the brain. And the data was collected while the patient was waking up after surgery after being subdued by propofol. And before we could begin analysis, the data went through a few pre-processing techniques. The first is we applied a bandstop filter at 60 Hertz to remove any noise from electrical lab equipment. And the second is we split the data across all channels into 30-second epochs in order for analysis. The next, the data was then transformed from the time domain to the frequency domain using the Welch's method. In the frequency domain, we're able to visualize the power spectral density in order to analyze the magnitude of the frequencies in the brain and how they change over time. In the collected signal from our electrodes, the collected signal from our electrodes consists of 5 major frequency bands as listed here. And for our case, we're concerned with the beta and gamma frequency bands as they correspond the most to conscious activity. So the first brain region we looked in was the parietal lobe. And here we see a decent distinction between unconsciousness and consciousness. So the unconsciousness is represented by the power spectral density at minute 20, which is a few minutes after the administration of propofol was stopped. And the consciousness is represented by the power spectral density at minute 58, which is right after the patient is able to keep their eyes fully open. Also in our power spectral density graph, I included minute 40, which is where the power spectral density represents the moments right after the patient starts to exhibit signs of limb movement. As you go down the brain towards the temporal lobe, here we see the greatest distinction between unconsciousness and consciousness. Specifically, we see large increases in the beta and gamma frequency magnitudes. This can be attributed to the relative close proximity of these electrodes to the brainstem and the thalamus. However, on the other hand, we do have electrodes, which we're typically seen on the outside, on the right side of the brain, have essentially no distinction between consciousness and unconsciousness. And this could be attributed to further distance from the brainstem and the thalamus. So after this analysis, I then trained a support vector machine and linear discriminant analysis models on the time and frequency domains of 2 data sets. One was the original data set, and the second was a new data set with noise removed and only keeping the channels where we could see distinction. So this means removing the channels like the electrodes from like the outer region of the brain where we don't see any distinction. And then I tested the accuracy of these models and compared them against each other. And here we can see that training the classifiers on the new data set shows improved accuracy across the board. Overall, the main takeaways is that we see the largest distinction in the power spectral densities in the temporal and parietal lobes. And by removing the channels with no distinction, we're able to have accurate classification models. And for the future, I would like to look at more patient data and be able to train the models on more patients in order to remove any biases that might come with just looking at one patient, as right now we don't have that much patient data. Also, I would like to try out different classification models and see how that turns out. And I would just like to give another quick thanks to the Grid Lab for hosting me this summer and allowing me to be part of this wonderful opportunity. Now I'd like to open the floor to any questions. Yeah, so this was I was a little confusing at first. Like looking at it, you wouldn't really think there'd be a difference in the left and the right side due to consciousness. I think one of the things is really was only able to look at one patient data, so we can't see how this how the behavior might be in different brains. But also, I think because on the right side, the electrodes, there weren't as many electrodes on the right side as there were on the left side, and those electrodes were farther away from the brainstem and the thalamus.
For your frequency graphs, could you explain the 3 dips that we commonly see in each of them? Is that sorry. That's the bandpass filter. So I applied one at 60 Hertz, but also the harmonics of 60 Hertz, like 120 Hertz and 180 Hertz, I applied that too because I was seeing spikes at those regions as well.
Okay, thank you very much, Josh. Our next presenter is Sarah Pierce-Lundgren from the Tuthill Lab.
Hello, everybody. My name is Sarah Pierce-Lundgren, as Eric Chudler so kindly introduced me. And today I'll be talking about some insights into proprioceptive function of the abdominal chordotonal organ and Drosophila melanogaster. Today I want to talk to you about proprioception and gaps in literature. What is the abdominal chordotonal organ, or ABCO as I might refer to it from now on? What is the structure and physiology? Where do the axons project to? And what is the connectivity and possible functions of ABCO? So proprioception is the ability to sense limb positions as we move. So the reason why I know that my arms are up in the air right now without even looking is because of proprioception. The Tuthill Lab uses Drosophila or fruit flies to study proprioception for a few key reasons. One of them is that we have the access to a large genetic toolbox. And what I mean by that is scientists have been breeding fruit flies for centuries, and now we have strains of flies so specific, we can label whatever we want down to the single cell level, which is pretty amazing. And the other key reason is that they don't have hard bones surrounding their version of a spinal cord called the ventral nerve cord. So we can image nerve signals integrating into their ventral nerve cord in a live, awake behaving fly, which is hard to do in other model organisms. Historically, proprioception has been studied in the limbs, but proprioceptors are all over the body. As you can see, this fly is wiggling its abdomen as it moves around on the slide treadmill. And it's just really essential to see proprioception all over the body, and not just the limbs and its relation to the main body. So this leads me to the abdominal chordotonal organ and what even is it? It was originally brought to the lab's attention because we commonly use a strain of fly called inactive gal4, and it labels or tags all of the proprioceptive organs or chordotonal organs in the fly. And the ABCO was also tagged in this genetic line, but nothing was really known about it by the lab or even by previous literature. So this is from the last known publication, really even mentioning or detailing the ABCO, and it was published nearly 3 decades ago. So what I did was I took updated images of ABCO and I manually counted the cells on each side. And what I discovered was that the amount of cells on each side was a lot higher than previously thought, with about 30 cells per side than the previously recorded 20 cells per side. Previous work in the Tuthill Lab discovered in the femur chordotonal organ that there were 3 different compartments. And so I did a staining to get a better idea of the structure of the ABCO. And what I discovered is that there also might be different compartments and that might have different functionalities. And they might be responsible for sensing different physiological sensations. So this finding was exciting because it indicated that the ABCO might have a similar structure to other chordotonal organs in the fly. So after I did that, I looked at an electron microscopy data set with computationally reconstructed neurons, otherwise known as a fly connectome, which is kind of like a map of all the neurons in the ventral nerve cord. And I identified 2 morphologically distinct types of axons. As you can see, there are these 3 boxes here. And each of those boxes is an area that controls each of the 3 pairs of legs in a fly. The first type terminates sooner in the ventral nerve cord in T2 and T3, while the second type goes all the way up in the ventral nerve cord and terminates in T1. So after I identified those axons, I looked at the downstream connectivity and at what those axons were, those axons were synapsing with. And I discovered that there was a high connectivity to interneurons that were associated with leg function or leg and wing function. But interestingly, there were no connectivity to abdominal motor neurons, which leads to a lot of interesting questions about the function of ABCO. I think it's possibly involved in postural control or flight control even. But yeah, I definitely have some cool ideas about testing those hypotheses in future in vivo experiments involving optogenetics and calcium imaging. And I'd love to talk to you about that if you come to my poster session. I'd like to thank the CNT, the Tuthill Lab, and my mentor, Ann Sustar. And that's it.
Questions for Sarah and Maddie?
So I might have missed this portion, but I was just wondering about the methodology of it. So you were saying that you connected the fly to some kind of like fly treadmill, or like what was the connection? Is it like some kind of probe that you're connecting to that you're able to see the signal? Or
So the work with the fly treadmill has more to do with my future directions. It was more so just to show examples of that. But so basically what I did was I utilized genetic and imaging techniques to get a better idea of like what the structure of of APCO is. And then I looked at I used computational methods to look at a data set that had the neurons, and I could examine the connectivity that way.
Is anyone in the lab recording from these neurons?
Not from these neurons specifically. The work in the lab mostly focuses on the femur cortitonal organ, but there is some electrophysiology work done in the lab, yeah.
We have time for a question or two more for Sarah.
Can you, I guess, look at that level of resolution on other neurobiology beyond flies?
There is work being done in the mouse, but mice are a lot more complex, and there's just a lot more data that needs to be taken into account when working with mice. Flies are just a lot smaller, and it's a much more of a direct process with a lot less data. So.
Last one, yeah.
Yeah, on that note, the simplicity, I was wondering if you could speculate differences between the type 1 and type 2.
So yeah, those were initially found because in the data set there are cell annotations made by another team. And so yeah, it was just really interesting to see them terminate in different locations because it might control different parts of the legs, for example, or maybe control different wing functions.
Thank you, sir. Our next presenter is Audrey Davis.
Hello, everyone. I'm Audrey Davis. I'm a senior at the University of Puget Sound, double majoring in economics and neurobioethics. So as an economics major, I've learned to know that everything can be used as a commodity, including our brain data. And as a bioethics major, I know that protecting the public, especially from privacy invasions, is paramount to maintaining trust. And so I've spent the past few months with the neuroethics lab here reviewing the existing literature on that intersection. And while it's hard to boil down everything from the past few months, I'm just going to hit a few of those key points, but with the end in mind, keeping that there's a long road to ensuring that there's a good data governance system implemented for neurodata, especially as it's so complex and broad and still developing. And so just to give a little bit of a background, neuroscience funding and in-hand innovation has exploded in the past decade. We know that we're going to be spending a lot more time here, but keeping in mind that most of the public at this point still doesn't have a great grasp on what neurodata is and how it can be used. Part of that is because we're still figuring that out ourselves. Also important to note that most people don't trust how their data is collected. Over 80% of US adults felt that they had little to no control over what information was collected about them from companies and believed that the risks of that data collection outweighed the benefits, which is important to keep in mind when we consider the increasing blurred lines between commercial and clinical boundaries. And so neurodata repositories are rather still in stages of infancy. So I used biobanking, which has almost 2 decades of literature examining their standards of data governance to do kind of assess the next best steps for neurodata repositories. Both hold similar privacy concerns. We see re-identification. So even though in research studies, or if you send something to a biobank or neuro repositories, all identifying details should be removed to maintain privacy, there is that threat of re-identification that exists. Secondly, there's consent. Do people understand what they're agreeing to, how their information will be used? Empirical studies already showed that most people don't have a great grasp on consent. And as this continues to evolve, that becomes an important question that ties to ownership. Who owns these samples, whether it's a tissue sample, blood sample, a brain scan, and anywhere that evolves from there. So these are potential sources for data misuse and ultimately a lack of trust if things go wrong. But biobanking does provide a solid foundational understanding for steps to create good governance systems. Importantly, we learned that you can't just have this system of governance from consent to collection to data sharing where data is protected, but you also need external safeguards like HIPAA, GINA, which protects companies and insurance from discriminating against your biological material. So not just protecting data, but preventing external harm. And of course, finding a line between responsible regulations and restricting innovation. And there are many ways to go about that. But as I've mentioned, the systems for neurodata are relatively novel, still immature, and they also present this idea of increased sensitivity. You know, DNA can tell you a lot about your disease history, your family history. Neurodata can do similar things, but there's also this potential of exposing behavioral patterns, disorders. As Tom Oxley refers to it, it's the last private part of ourselves. We see here below Synchron's first brain-computer interface assisted tweet from paralyzed Philip O'Keefe. So we've seen the positive that can come from these novel neurotechnologies. On the other side, we have seen the abuses that may come. So a lot of these popularized news outlets running headlines about your big brother watching, Orwellian kinds of things. There's both good and bad, but we do have to recognize that as these questions of cognitive liberty, do you own your thoughts? Do other people have access to them? That these are the kinds of headlines that most people are reading rather than all of the scientific studies that we publish that show the good. And as legislators kind of step in to acknowledge what needs to be done, it's important. I'm just going to show a quick clip to remind us that at the end of the day, politicians are largely still just lay people.
Well, so how do you sustain a business model which users don't pay for your service?
Senator, we run ads.
I see. That's great.
If I'm emailing within WhatsApp, does that ever inform your advertisers?
No, we don't see any of the content in WhatsApp. It's fully encrypted.
Let's say I'm emailing about Black Panther within WhatsApp. Do I get a WhatsApp? Do I get a Black Panther banner ad?
Senator, we don't. Facebook systems do not see the content of messages being transferred over WhatsApp.
Yeah, I know, but that's not what I'm asking.
So important to keep in mind that while legislators are taking steps to reel in big tech and make regulations, that they don't necessarily understand all the science and technology. So this requires engagement from all areas, not just legislator and industry, but science, econ, ethicists, to ensure that there's responsible and efficient data governance. And we have these private or explicit safeguards for our genetic material, but we still haven't developed things for neurodata. So analyzing privacy models to ensure that we can have regulations and safeguards to protect neurodata. And of course, bringing in end users, having empirical studies, engaging the people who are going to be most impacted, which at the end of the day could ultimately be all of us. So thank you.
Well, so how do you?
Special big thanks to the Neuroethics Lab and all the support from everyone there.
Questions for Audrey? Raj?
Yeah, the product brings up really interesting questions, especially the differences between biobanking and neurodata. And one thing that I always wondered about is, with the neurodata, especially with the new generative AI type techniques, right? You can actually take the data and you can compress it, so to say, within a model, the generative AI model. And then I guess the question becomes, it's not really, can they claim that that really isn't your data because we now sort of amalgamated or compressed into an AI model, right? But then it's generative, so it could reproduce original data to some extent, right? So I think there's some really interesting, tricky issues that we need to confront. So I don't know if you had thought of that.
Yeah, I've definitely been thinking about that a lot, especially with the idea of consent, because in most commercial senses, we click agree to selling our data all the time in that sense, whether we're actually agreeing to it, but that's kind of their safeguard against legal ramifications. So I definitely see a lot of need for work and conversations there. Yeah.
Great, thank you. Our next presenter might have some additional comments, perhaps about consent. That is, our presenter is Noah Long.
All right. My name is Noah, everybody. Thank you. So the work that I mainly am doing is actually analyzing a new generation of brain pioneers. And so I'm just going to kind of do like a quick overview of the ethics, specifically identity formation and the introduction of children in neurotechnology. And thank you for the Gehring Lab. So first, let's just do like a brief definition. Brain pioneers are kind of like people who volunteer, they take risks, they offer their time, their efforts, etc. in studies and in cases and such of brain technology, and trying to uncover the secrets of how we can improve quality of life. So a main research question that came out of that for me was how should children actually be integrated in such complex and ethically confounding neurotechnological research? And so hopefully you guys take this journey with me as we kind of do some speculative work here. So there's actually multiple ethical complexities that can come up, but I do want to kind of mention that there actually is a disconnect when there comes to parents, clinicians, and children. And so parents typically kind of want what's best for their kid. They want increased quality of life, etc. Clinicians, sometimes they do, but mainly they are very interested in promoting research, and there is a desire to help, but at the same time, there's a disconnect that exists with the parent and children, in which children are also very interested relationally in forming connections with others, and then on top of that, increasing their own, I would doubtfully say quality of life, but rather increasing connectivity to what they can and can't do. And so that actually kind of brings in like the idea of like accessibility issues. And so it's kind of very, very important that not only is consent given not only by the parents or the adults in the situation, but there also has to be functional assent by the children in the study, because if they are not necessarily made aware of all of the complexities, which they might not understand, then this could cause further issues multiple times down the line, because these are studies that can last like 3-5 years, etc. And so a lot of the current child pioneers that we have are kind of having existing conditions such as dystonia, epilepsy, tetraplegia, etc. And so a lot of these are actually classified as physical disabilities, which are very separate from mental, which would be like OCD, depression, etc. And so currently, as it stands, a lot of this technology is not being kept. And so there's an issue as it comes to the removal of the technology and what this actually means for the child. And so they've been a part of all of this technology. They've been a part of all of this groundbreaking work. But now there's an issue with almost an obligation, obligatory question of whether or not we should continue to allow access to technology that actually benefits their life. And so I kind of just want to bring up future directions in this, because right now it's very, very speculative. So with all the literature that I've been reading and thinking about, there's some three like very, very crucial ones that I put here. And so what's really, really important, pertinent, is the necessity to analyze not only children, but what the technology that they currently have, but also the technology that they are almost being stripped of as these cases and these treatments are terminated. What does this do to the child? What does this do for their identity? What does this do for their relation with their peers? What does this mean for their quality of life? What does this mean for anybody that's involved? And so these questions are questions that we're working on currently answering. But what's a main takeaway for me is this is really important work. This work is something that does need to be undergone. It does need more research and hopefully more literature. And I think that if you guys join me at my poster, we can do some nice speculative work together. Thank you guys for your time. And I want to give a special thank you to Dr. Chutler, my lab, the neuroethics lab, and also everybody who's been a part of this. Thank you for this opportunity.
Questions for Noah?
During the literature review, did you find any discussion about different age groups of children? You know, because I know a lot of neurotechnology is going to be focusing on like early intervention. It's going to usually be, you know, more neuroplasticity, things like that. So I'm wondering if it was like, do we look at a newborn to 2-year-old different than a 2, 4, 5, you know, school age kids?
So actually there is a little bit of a divide in that sense, because when implanting technology, it has been seen to be safer in children who are younger. And so before the age of 2, it does typically lower the cases of infection. And then also older than 7, you see a little bit more infection, but also 7 and the rule of 7s is like an identity formative period. And so there's very tentative work that kind of gets done there. But that is an interesting question, because there are very clear divides as it comes to when it's, I guess, necessary or applicable to implant this technology.
Okay, thank you.
Thank you.
We have one more presentation before another short 5-minute break. Anthony.
Hi, my name is Anthony Capozano, and today I'm going to be presenting the research that I did at Stuber Lab over the summer. And what the research that we did was dopamine signaling across the striatum during consumption. And some background on the striatum is the striatum is involved in decision-making and motor control and some emotion and reward. And some other background about dopamine is that dopamine is critical for learning. And we see that when we're looking, what we're going to be focusing on are the midbrain dopamine projections. And we see that the midbrain dopamine cells, so we know that the midbrain dopamine cells are critical for motivated behavior, and individual dopamine cells project to discrete brain areas. So we see that the tail of the striatum is receiving inputs from the globus pallidus, the subthalamic nucleus, and the endopennuclear nucleus. However, when we see the ventral striatum project, we see that the tail of the striatum the midbrain dopamine cells that project to the tail of the striatum receive a unique set of inputs relative to the other parts of the striatum. And when we look at the initial connectivity matrix ex vivo, so this is just the connectivity patterns, we see that the correlation, if we focus right here on the tail of the striatum, has less correlation compared to the other brain structures. And the methods that we use, what we were trying to address is the signaling patterns across the striatum with motivated behavior, which is during consumption, and without motivated behavior. And we want to see whether or not if all the brain regions respond the same way during this behavior. And so when we take a look at the methods that we use fiber photometry, which expresses light and stimulates the animal's brain. And we see it using the fiber photometry. What we injected into the mice was GrabDA, which is a which is a protein that changes fluorescence based on the dopamine level. And what we're going to see is the dopamine release across the animal throughout in the midbrain and the striatum. And we're going to see what that looks like throughout. And what we used for the motivated behavior is the animals were head fixed, and what we used to assess the dopamine levels during motivated behavior is the mice were water restricted, and we used sodium chloride, which is just salt, which is really highly aversive to the animal. And in return, what the animal would do is they would lick, and if you can see right here, this is just water, which is a lot more highly rewarding to the animal. So this is going to be it licking a lot more. And then when we take a look at the sodium chloride, they lick for a little bit and almost immediately stop licking. So what we expect to see here is that the dopamine release should be a lot more lower with the highly aversive solution. It should be a lot more higher with the water because it's a lot more rewarding to the animal. And also a lot more less licks with the highly aversive solution. And when we take a look at the results of dopamine signaling across the subregions of the striatum, we see with the raw signaling data right here that the fluorescence is the y-axis. So this is just the brightness level of the dopamine that's being released over the time interval. We see already that the initial results show that the tail of the striatum has different signaling already compared throughout the rest of the striatum or the subregions of the striatum. And we take a look at the baseline combined. This is just the correlation matrix of all this data put right here and simplified. We see that the results show that the dopamine release in the subregions of the striatum are correlated with neighboring regions. So if you see all of this red right here, the nucleus accumbens right here is correlated with the dorsal medial striatum, and as the further it gets, it starts becoming less correlated. And the other interesting result that we found was the dopamine release in the tail of the striatum is negatively correlated with the nucleus accumbens right here. As you can see, the scale right here, the red being a lot more correlated and the blue being less correlated. It's negatively correlated with the nucleus accumbens. And we look during consumatory behavior of the sodium chloride solutions and the scales of the striatal dopamine. What we see is we have the fluorescence again on the side and we have the seconds on the bottom. We see with the higher concentrations of sodium chloride, it shows the consumption shows a decrease in dopamine in higher concentrations across the striatum. And we see right here with the average of the fluorescence, we see already with each individual test subject right here that the tail of the striatum shows almost no scaling compared to the rest of the subregions of the striatum and the nucleus accumbens. And we take a look at the consumatory behavior and we see this with the correlation matrix right here during consumption. We see that during consumption, the tail of the striatum shows little correlation with the neighboring structures. So as you can see, a lot of the structures right here in the bright red are a lot more correlated with each other during consumption, while the tail of the striatum has little correlation with itself, with the other neighboring regions. So for summary and future directions, what's important is if we take a look at the initial connectivity matrix right here, we see that the correlation with the darker being a lot more correlated. We see that the tail of the striatum initially has little correlation, and we see this during consumatory behavior. And we see that the tail of the striatum has little function relative to the rest of the striatum during consumatory behavior. And some limitations of our experiments they were conducted in a small cohort of mice. We used 4 mice, and we will need to increase the sample size before making any conclusions. So some future experiments that will accomplish this is stimulation or blockage of the dopamine in the tail of the striatum to see what may have a causal role during consumption. And so this data is going to help us understand how dopamine is released across the striatum during reward and aversion, which has applications for how dopamine coordinates food consumption. And thank you. I would like to thank the Stuber Lab and everybody that showed up, and thank you for the opportunity to present my research. Thank you.
Time for one quick question for Anthony.
Here's some further specifications that I included in my research too. So I mean,
okay, thank you.
Thank you so much.
Okay, we have another short break. We're going to start again promptly at 11 o'clock, so please don't go too far.
I see a water bottle.
Oh, yeah, those are for you. Yeah, absolutely.
Oh.
You give my nephew a break. I'm just kidding, of course. Let's get to the wall. I love it. Shorter people.
Dr. Tread.
Thanks. Are these working? Should I use that or should I just?
This one's fine. Okay, if everyone could take their seat again, we'll get started. I'll just do a quick reminder too. And just as a reminder, we have one more block of talks, and then there'll be an hour break between about noon and 1 o'clock. And on your seats or in the back, there's also a QR code that has the program as well as everyone's abstract. So you can go ahead and download that program if you like. Again, just in the room next door from 1 to 2 o'clock, we'll have half of the poster presentations. And then from 2 to 3 o'clock, we'll have the other half of the presentations. The next talk is by Ryan Wang from the Osborne Lab.
Hello, everyone. My name is Ryan. I'm an undergraduate intern at the Osborne Lab with the Center for Neurotechnology. And today I'll be presenting my work on the neural decoding of primate reach direction in brain-computer interfaces using a novel machine learning model. So for a little bit of context, what is neural decoding? Essentially, it's taking the signals that we measure from the brain and translating them into what they might mean or what the intention of the person was. And this is very useful in the context of neuroscience, where we might be trying to figure out how the brain is working, and in the context of neuroengineering, where we might leverage these signals to develop tools to help with our capabilities. Now, the most common method for neural decoding is called machine learning, and this essentially uses artificial intelligence to recognize patterns in the neural signals that we collect and identify what they might mean. The most common method for neural decoding with machine learning is called supervised learning, where we take a set of neural signals and also a set of labels corresponding to those signals. This way, a neural network is able to learn correlations between the patterns in the signals and the labels, and is able to apply that to new data and signals that we collect. However, acquiring these labels can be time-consuming, costly, and also is prone to bias and human error. So we might turn to a different method of unsupervised learning, which does not involve the labels. Here we have a method called unimodal decoding with self-supervised learning that takes the neural data on its own and kind of generates pseudo labels with the data. In this way, it's able to classify the neural data without the need for a set of true labels. In order to improve the accuracy of this method, we might turn to what's called cross-modal decoding, which leverages other streams of data that might be highly correlated with the labels that we desire for the neural signals. For example, we might take the neural data and also kinematic data like hand movement data to figure out what neural signals are correlated with different types of movement. In this way, we're able to generate more accurate labeling with unsupervised decoding using multiple data streams. Now, this cross-modal self-supervised method has been implemented in tasks such as detecting whether an arm is moving or not, and also detecting which finger is moving. In our case, we might want to implement a more complex decoder in a task that involves moving a cursor from the center of the screen to one of 8 different directions. Another thing that we want to investigate is what streams of data are most useful for this task. So we'll investigate eye tracking and kinematic hand position data to see whether that will help inform the self-supervised decoder. In order to do this, here's this experimental setup that we have. Essentially, we have a non-human primate completing this center out task where it moves a cursor from the center of the screen to one of 8 different targets. At the same time, we record from 3 different streams: electrocorticography, which is the neural signals, 3D hand position using motion capture, and 2D eye position using an eye tracking software. These 3 streams of data are then fed into the neural network to learn and correlate different signals. We also take a portion of this data to a test corpus so we can test the model after it's been trained and learned on the data to figure out and evaluate its accuracy and performance. The model can then take these neural signals and predict a decoded reach direction given the 3 streams. Here are the results that we have from several of the models that we trained. Our first primary result is that the cross-modal models, which utilize streams such as hand data and eye data, without exception outperform the unimodal model, which uses just the neural data. This tells us that the eye data and the hand data is informative in the self-supervised and unsupervised learning paradigm. Another thing that we find is that these cross-modal models' performance approaches the accuracy of the supervised learning model, which is the gold standard. And that's something that we look to improve, is the accuracy of these models to approach that of the supervised model. When we look more closely at the decoding performance of these different models, we see the following patterns. So here for the supervised model, you can see it performs very well on the training data. When the actual target is target 1, it often predicts target 1. When the actual target is target 2, it often predicts target 2, et cetera, and hence you see this dark green diagonal line. With the self-supervised model on the training data, you can see that it's able to interpret the signals pretty well as well. You can see a pretty clear green line down the diagonal. However, on the test data, when we apply this to data it hasn't seen before, it has a little bit more trouble generalizing these patterns. And so what we hypothesize is that this model might be a little bit overfit. In other words, it's very good at recognizing stuff with the data that it was trained on, but not as good as applying it to a broader context. Another thing that we find is that with more data, we have increased accuracy. So for the supervised model trained on a day with more trials, you can see a darker green line compared to a day with fewer trials. In conclusion, we find that multiple data streams does increase the accuracy of the self-supervised decoder compared to the unimodal, and that self-supervised decoding could be improved in various ways. We also, if this is successful, would want to implement this with more complex tasks, which may not include data labels, such as with brain-computer interfaces. I'd like to thank my lab members, including Dr. Orsborn and Catherine, for their support and guidance during my experience here. And I'm open to questions. Thank you.
Questions for Ryan?
Great talk, Ryan. Take us into the future and tell us how you think this might work with someone at home using a BCI that has to be recalibrated every day.
Yeah, that's a great question. So typically, maybe with supervised learning, we might have to have a lot of labeled data to calibrate the brain-computer interface. In this case, it might be something as simple as just, waving your arms around, and that, would be a lot simpler and take a lot less time than having to, choose specific targets and specific labels to assign to the neural data. So that might be a lot faster and a lot more generalizable and accessible for people who might want to implement these brain-computer interfaces.
One quick question in the back, yeah.
These models are specific to the person, or could you train models on subset people and use it to?
That's a great question. So I think it will depend. I think if we were to generalize this to a large population and use different people's data, it would create a good baseline. And for each different person, we could start at that baseline maybe, and then have it be more specific, like a starting point, and then make it more specific per person.
Thank you.
Yeah, you can see me at my poster for more, if you have more questions.
Our next presenter is Allie Williams.
Hey everyone, my name is Allie, and I've been working in the Cognition and Cortical Dynamics lab with Dr. Stocco this summer, and I'm going to take you through my presentation, the optimization of a computational model for diagnosing mild cognitive impairment, or MCI. So mild cognitive impairment is a memory classification describing people who have abnormal memory decline but are not yet classified as Alzheimer's or dementia. But it's one of the highest risk factors for the development of dementia in the next few years. And early intervention and treatment have the best chance of helping patients. So MCI is currently diagnosed during a clinical evaluation with a full neuropsychological battery, as you can see here. It takes around 50 hours to administer for an accurate diagnosis. Most notably on this list is the MoCA, or the Montreal Cognitive Assessment, which is a pencil paper test that's currently the gold standard for assessing memory decline. This process is hindered by clear limitations, as it's expensive and time consuming for the patients, and it can only be administered once a year due to practice effects. So to remedy this, our lab created a new way of assessing memory decline. And it uses an adaptive learning software to introduce new fact pairs and ask participants to retrieve them again later, just before they're predicted to forget them. So groups of MCI and age-matched healthy control participants in a prior study did 8-minute learning sessions, such as these, weekly over the course of a year for a variety of lesson topics such as birds. The learning system was developed using these equations from a popular cognitive architecture modeled after human declarative memory. They interact to tell us the activation of a memory M at time t based on the accuracies and response times collected during the task. This leads us to estimate the alpha parameter, which tells us the speed of forgetting. Speed of forgetting is a measure of individual differences and long-term memory function. And this is what we're actually using to estimate somebody's learning or memory ability. For example, and to contextualize this, an undergraduate with a speed of forgetting of 0.3 in that green line has a 5% probability of retrieving a learned fact within 6 hours, whereas a dementia patient with a speed of forgetting of 0.53 might remember that or might forget that learned fact within 5 minutes. So speed of forgetting has also been shown to be stable over time, as the MCI patients in that purple line have a distinctly higher speed of forgetting over time than our healthy controls in the yellow line. For analyzing its diagnostic capability, we can look at an ROC curve, which just shows the true positive rate and the true negative rate for classifying MCI versus healthy controls. And we can see that the gold standard MoCA is currently at a 93% classification, whereas our speed of forgetting model can get to an 87% in only a single 8-minute session. So that brings us to my project, which is optimizing the model to improve that 87% classification accuracy. So currently, the model assumes that participants have no prior knowledge for any of the facts introduced during a session, meaning that the base level activation for a fact is currently set to 0. However, this is not the case, as many people may be familiar or even know facts ahead of time, which would bring up the base level activation and make our calculated speed of forgetting values inaccurate. So this summer, I designed a study that would assess prior knowledge ahead of time. And 20 undergraduate students took a survey and then completed 2 learning lessons, Asian flags and Caribbean flags. I figured this would provide a wide variability of people who knew certain flags ahead of time, as I'm sure many of you could tell me this top flag is from China, but not as many could tell me that the bottom flag is from a Caribbean country called Anguilla. So the first step was to test if prior knowledge was affecting our current speed of forgetting estimates. We saw that for each participant, the facts for which they had prior knowledge in green were significantly lower than the facts that they did not know ahead of time in blue. And a mixed level model could confirm these results. This means that the model is incorrectly estimating that they're better at learning these facts, but they really just knew them ahead of time. So to fix this, we used maximum likelihood estimations, or MLE. This is a method we have not previously used before that incorporates trial by trial likelihoods to choose parameter values so that the model maximizes the likelihood of producing the observed data. Using MLE, we were able to accurately recover the base level activations and then use that to infer whether or not they had prior knowledge. And we were right about 69% of the time. While there's room for improvement, this means that including prior knowledge into the model will improve our classification accuracy. So to begin analyzing our previous clinical data set with MCI patients, we can see which facts have abnormally low speeds of forgetting. And you can see that for these facts, they are correlated with the facts that have the highest base level activation. This means that retroactively, we can tell whether or not patients in our clinical data set had prior knowledge. So then we can go in and correct for that. So the next steps will be to do this throughout the entirety of our clinical data set. And I then want to estimate other parameters in the model by implementing an eye tracking study that I have for this upcoming fall. So thank you so much for everyone in my lab who helped me tremendously with this project, and I couldn't have done it without you all. Thank you so much.
We have time for one quick question for Ali. Jeff.
Okay, well, but I'm not asking anymore, so somebody else has to step up. In your intro, you had that, I think it was like 52 weeks of stable speed of forgetting, the alpha variable for the people with cognitive impairment. I noticed the healthy controls were trending down, which sounds familiar as I age. What do you think was going on there?
Yeah, so just people normally decline over time in their memory ability. So like healthy undergraduates have a speed of forgetting of 0.3, but then our age matched like healthy controls have a speed of forgetting of about 0.37. So people just naturally get worse over time. And so actually the initial goal of that study was to track memory decline. So we see just natural progression in memory decline, and we're seeing who is declining more, whether it was the MCI versus the healthy controls. But that was a great question.
Thank you. Our next presenter is Maria.
Okay, so good morning, everyone. My name is Maria Vasquez Rivera, and today I will be presenting the project we did during the summer, estimating the effects of ischemic stroke and stimulation on the connectivity between brain regions using graph learning. So among the different types of strokes, ischemic strokes are the most common. They occur when a block clot blocks an artery in the brain and cuts off blood flow and oxygen supply to that area. Now, this may cause disruptions in brain network activity, which often lead to lifelong disability. And while in recent years, therapies such as brain stimulation have shown promising results in treating post-stroke symptoms, our limited knowledge on how strokes and subsequent stimulation affect neural activity is hindering progress. So the purpose of our study was to investigate the effects of ischemic stroke and subsequent stimulation on brain network connectivity. So for the experimental setup, in a previous study, colleagues induced focal ischemic lesions on 4 non-human primate models. And they did this using the photothrombotic technique, in which they illuminated through an opaque mask using a white light source, as can be seen in figure 1. Then they captured images of the vascular networks of each NHP and saw clear cortical blood flow disruptions as seen in figure 2. They also used a semi-transparent 32 electrode ECOG array in order to record and stimulate neural activity, as seen in figure 3. Now, with this recorded data, we were then able to do our connectivity analysis. So first, we plotted a time series graph of each channel in the ECOG array and put this through a new graph learning algorithm. Now, this algorithm estimates networks from data by solving an optimization problem that finds a graph such that the neural activity between connected nodes is similar. And so we then took the learned data from this produced graph and plotted connectivity networks for the before stroke, after stroke, and after stimulation instances as seen by these example plots. Then we were able to calculate the average change between the before stroke and after stroke instances and after stroke and post-stimulation instances and plot difference networks for the ipsilational and contralational hemispheres, displaying changes in connectivity as shown by these example plots over here. Now, when looking at the difference networks for the ipsilational hemisphere, we saw a mix of post-stroke connectivity effects across all NHPs. And we see this by the inconsistent patterns of blue, white, and red edges between nodes in all NHPs. Then, when looking at the difference networks for the contralational hemisphere, however, we saw an increase in connectivity post-stroke. And these networks were computed by calculating the average change between the before stroke and after stroke instances. And if we compare the post-stroke effects between the two plots, we see a clear increase in connectivity as denoted by the vast amount of red edges in the contralational hemisphere as opposed to the ipsilational hemisphere. And finally, when looking at the post-stimulation difference networks, we saw an increase in both the ipsilational and contralational hemispheres. Now we computed these networks while calculating the average change between the after stroke and after stimulation instances. And if we look at the both networks, we see a vast amount of red edges, which indicate an increase in connectivity post-stimulation. So from this study, we gathered that there was a mixed post-stroke connectivity effect in the ipsilational hemisphere across all NHPs. We also saw an increased connectivity in the contralational hemisphere post-stroke, which may be as a way to compensate for the reduced connectivity in that of the ipsilational hemisphere. And finally, we saw increased connectivity in both the ipsilational and contralational hemisphere following electrical stimulation post-stroke. Now, as for future works, we would perform a more rigorous statistical analysis of the data, and we could also analyze further brain regions by using a larger ECOG array. Now, I would like to thank my mentor, Felix Schwach, for taking me under his wing during the duration of the program, Dr. Destam for allowing me to be a part of the Nerd Lab this summer, and all the members of the Nerd Lab for making this such a memorable experience. Thank you very much, and if you have any questions, please do not hesitate to ask.
Questions for Maria?
And so you mentioned that on the contralateral side, there's an increase in connection kind of after the stroke.
Mhm.
And one of the possible reasons is to compensate. I was curious whether there were any controls, like is that something that could also occur naturally? And then if maybe there are other reasons that you might hypothesize for why there's such an increase.
Well, I'm not sure if there is like if that's something that naturally happens. I'm pretty sure like it doesn't, but we just really thought that it was, wait, let me go. We just saw it as it was a way to compensate for the reduced connectivity in the educational side, which also goes along with the literature that's already out. So.
Raj?
Yeah, so, in terms of showing the connectivity between before and after, right? So what you'd like to do is show there's a consequence on the recovery after stroke, right? So was the connectivity measured during a task or was it during resting state? Can you explain a little bit about how the connectivity measure was extracted from the brain?
Well, from what I was told and what I read, I know they did post simulation for, I think after an hour that the stroke occurred, and they did 1 hour of post simulation and then recorded the data for 3 hours. But I'm not sure if it was during a task or if it was just like them resting. I think maybe it was them just watching videos, but that may be, I'm not sure about that.
Okay, thanks, man. That's really good.
Thank you.
Thank you, Mary. And there are some more chairs up in the front if people want to fill these up, and there's a few chairs in the middle as well if you want to take a seat. And I'll let you set this one up. Our next presenter is Lucas, who is in the Riddell lab.
Maybe. Interesting. This didn't happen during testing. One moment. There we go. When in doubt, close and reopen the program. Hello, I'm Lucas Ritzdorf. I had the opportunity this summer to work with the Future Analog System Technologies Lab on a project that uses machine learning assistance to accelerate self-interference cancellation in full duplex radio transceivers. Seriously? One moment. Okay. First, an introduction to radio systems, hopefully without any more Acrobat glitchiness. This research focuses on in-band full duplex radio systems. That means that A, the transmission and reception occur on the same frequency band. Some systems use different frequency bands so that they can operate simultaneously more easily. In-band does not do that. And then the full duplex component means the system both transmits and receives at the same time. This is particularly challenging because of an issue known as self-interference. Essentially, the system will re-receive its own transmissions in an unanticipated or unhelpful manner. There are two ways that this can occur. First, there's direct coupling, where simply by virtue of having two electronic circuits near each other, the transmitter and the receiver, they will be capacitively, inductively, or sometimes even both coupled together so that the transmitter circuit directly affects the receiver circuit. That is typically, happens within about 1 nanosecond. It's a very short delay. The second vector is through environmental reflection. It's essentially echoing of the radio signal. The transmitted signal bounces off of elements in the surrounding environment and is then re-received after typically a much longer delay, although again, that is heavily dependent on the details of the surrounding environment. This is a very much simplified block diagram of the system that we're working with. There's along the bottom here a standard transmitter system with an antenna. receiver system at the top here. And then in the middle is the filter set. The red block is for direct coupling suppression to reduce the first type of self-interference that we've just mentioned. And then the orange block is a tunable finite impulse response filter that is controllable to properly compensate for environmental reflections. And that control is accomplished by some circuitry running on the green block there, which is a field programmable gate array, or FPGA. FPGAs are really, really neat, actually. They're special chips that can reconfigure themselves based on a digital circuit design to become an arbitrary digital circuit, essentially. That can be as complex even as something like a modern computer processor or a neural network. Before we get to that, though, this is the physical setup for our system. Again, the same general components are labeled here. The key components are the FPGA under that fan and heatsink, and this custom designed chip that our lab built as part of a previous project. But this is where the filtering step actually occurs. There are a variety of ways at the moment for us to calibrate this filter, but calibration is, as I mentioned before, important in order to properly compensate for reflections from the environment. As the environment changes, so will the reflections, and so must the filter tuning. And the faster we can do this, the more of a real-time responsive system we have, so the better performance we get. Our previous method for doing this was a pseudo-blind parameter search, where we essentially take the approach of an unprepared student on a multiple choice test. We guess and check by varying, in this case, individual bits of the tuning parameters, but we make a bunch of guesses, pick the best one, and then vary a little bit from there until we eventually converge to a well-calibrated system. The new technique is machine learning assisted parameter prediction. This is substantially faster. We'll get to that in a moment. But its quality does depend on the quality of the machine learning model for obvious reasons. With the model itself, we've had this mentioned actually in a few previous presentations. This is a convolutional neural network. It takes the later stages of a traditional neural network and precedes them with a convolution layer, which is essentially a way for the model to do its own feature recognition and extraction. It's typically used on graphical images, and it's also a lot easier to explain in that context, but it's good for general pattern recognition applications and should be useful here as well. This model is implemented in two ways for a project, one in software via MATLAB for the training process, and two on hardware in the FPGA chip for the actual system operation stage. Now, that performance I mentioned. The blind search algorithm takes just barely under 30 seconds to converge to a decently well-calibrated state. The CNN hardware model takes less than 32 microseconds, including the surrounding memory relay circuitry, to actually send those parameters to the filter itself. Less than 32 microseconds to make a single prediction. Again, since this is dependent, the results are dependent on the quality of the model, it might take multiple iterations of the CNN to properly converge to a tuned filter state. But if a single run takes 32 microseconds, we have quite a lot of room to mess around with running the model multiple times if necessary. That's also an improvement of nearly 6 orders of magnitude time-wise, which is not something you get to claim frequently in science in general. So that's been pretty fun. And then applications for this system, the generally accepted threshold for achieving real-time adaptation is retuning the receiver once every few hundred microseconds. Again, yeah, since we can retune in 32 microseconds, we can very much meet that threshold. One potential downside of our current system, obviously as a research prototype, it's not optimized for portability. That's something that A, comes with making a system that's not just a prototype, and B, given our lab's experience with dedicated chip design, there are potentially some interesting avenues to explore there. It would be substantially more complex and different from most of the analog designs they've done before, but something along the lines of a dedicated neural network chip might be an interesting option for future exploration. With that, I'd like to thank my project mentor, Po-Hao Cheng, our PI, Chris Rudell, assistant mentors Yisong and Chi Chen, and of course, the RU staff and students. Are there any questions?
Time for one very quick question for Lucas.
Can the system receive signals when it's calibrating, or is there a blind spot every time it's calibrated?
It should be able to operate nearly continuously. The calibration process is essentially a background step, and that's handled by the hardware on the FPGA. The transmit and receive ports on the system's output, which is where you'd hook up your actual data inputs and outputs, those should be functional the entire time, yes.
Thank you. Our next presenter is Brandon Fisher.
Hello, my name is Brandon Fisher. Today I'll be telling you about investigating molecular strategies targeted at genetic manipulation of function-specific neuron population after spinal cord injury. So here in my lab, we try to utilize therapeutics with spinal cord injury because many times spinal cord injury does impair significant sensory motor functions, and this also diminishes the quality of life. And so here we understand through prior research that in order to improve therapeutics, we must understand the functional recovery of therapeutics and how this hinges on circuitry rebuilding and synaptic reorganization. And so here we see that oftentimes in certain therapeutic studies, such as this right here, we see that the forward-reaching task has a higher success rate than the steering task, and they're both being optogenetically stimulated. And so here we're really trying to find out as to what neurons are being stimulated in the forward-reaching task to be able to help it to have a higher success rate. And so here in this study, we're utilizing the AV vectors to allow for labeling of active neurons in time-sensitive manner. So an example of this, so what I just said, is that we're trying to label specific neurons to support specific tasks, such as the FRT task. And so here, when we inject the AV vector into the left side of the tissue of the spinal cord, we then give them Doxchow so that it can inhibit transduction of active neurons. And before they actually get ready to perform an IBB task, or what I said prior, the serrating task, we then take them off doxylamine chow, and then we allow them to do the activity, and then we expect to see some neuronal labeling. And the reason why we want to use Doxchow to prohibit the active labeling of neurons is because we don't want any non-specific expressions of CDH2 during control activation. And so furthermore, we would investigate the time sensitivity of labeling active neurons by removing Doxchow while they undergo electrical stimulation, perform IBB, or rest in their cages. And so here in the experiment, we give them spinal injection, and then we give them Doxchow for 4 weeks. And then after 48 hours, we remove Doxchow as a performing activity. And then after 24 hours, we remove Doxchow again so that they can be perfused and have tissue processing. So here in the experimental groups, we have the first rat where they received no Doxchow and they got 1 hour IRB training. We expect a lot of moderately labeled neurons from this. And number 2, they received no Doxchow and they got 10 minutes of electrical stimulation, so we expect a lot of dense labeling. And number 3, they received no Doxchow and they also received no training, so we expect minimal to no labeling from this. And number 4, they received Doxchow throughout the experiment, and they also received IB training as well, so we expect minimal to no labeling from this as well. And so here we want to actually see if the new vector is able to label neurons in a time-specific way. So here in the experimental data, we have the experimental side and then you have the control side. So here we're basically trying to see this side, the experimental side, is the side that got the viral injection. We want to basically compare the two sides and see the neurons on both sides. So here, this is the rat that got the Doxchow removal, and they had IB training for 1 hour. So here you see a lot of moderately labeled YFP neurons. And over here is the rat that got the Doxchow removal, and they got electrical stimulation for 10 minutes. So we see like a lot of YFP labeled neurons are densely labeled. And here, this is the rat that got the Doxchow removal and they're resting in their cage. So you don't see that many YFP labeled neurons in the one down here in the bottom. This one got Doxchow throughout, and they also got IB training. So we kind of see some YFP labeled neurons that we expect from this that the rat can get enough Doxchow. So here in this tissue, we basically show that CDH2 can be expressed in electrical stimulation. and the wide label neurons can be optogenetically stimulated in future studies. So here, an example, the AV vector and the doc shell can potentially label test-specific neurons, which we see right here that I previously showed. And we also want to do this study again to understand why the wide label neurons in the control were there. And we also found out that CR2 in wide labeled cells can enable future task-specific neural activation. So for future plans, we want to repeat the study with more rats, maybe track how much doc shall they eat. We also want to study the AV vectors in labeling of active neurons and taxamorphin for receptivity, so a different vector with a different task. And so here we also want to study biovectors with spinal cord therapies for enhanced multitask recovery. And here again, I want to thank Dr. Mondello and the people in my lab, Dr. Chip Moritz, Viet Dang, and Rich Henderson for mentoring me and making this experience a very fun one.
We have time for one quick question for Brandon. I just have a quick one. I've never heard of DoxChow before. Do you know, can you tell us how it works?
Oh, so DoxChow, you can inject it, and also you can use it as like a food. So basically it's supposed to inhibit certain neurons, because if you just inject the viral vector and you don't do any sort of thing to inhibit it, it's sort of like you'll get like a lot of static. So you need to, but we want to label specific neurons, so that's why we use DoxChow.
Okay, thank you, Brandon. Our next presenter is Amina.
Hi, my name is Mina Alzatma, and this summer I've worked in the STEEL lab, and my project is called Take a Step, the effects of transcutaneous spinal cord stimulation and exoskeleton use on step length for children with cerebral palsy. So cerebral palsy is a classification of brain injury or lesions shortly before or soon after birth. It's the leading cause of childhood onset disability, and it affects approximately 1 in every 500 children. And CP can cause muscle spasms, muscle contracture, rigidity, overall weakness, and in some cases paralysis. In relation to having trouble with moving, people with cerebral palsy often have shorter step lengths. Children who have shorter step lengths, step length is defined as the distance between the initial point of contact of one foot to the initial point of contact of the other foot, as can be seen by the diagram in the bottom right corner. The consequences of shorter step length are depleted energy levels, lack of physical activity, and an overall decrease in quality of life. Traditional interventions targeting improved walking in children with cerebral palsy include treadmill training, walker training, and orthosis usage. These devices have mixed effects on increasing step length for children with cerebral palsy. So we are investigating technologies that increase engagement during physical sessions to hopefully increase step length. The first of these technologies is a biomodem ankle exoskeleton, as can be seen up here. It's comprised of a battery pack on the participant's back, an ankle pulley, a force sensor in their shoe, and we have an app that allows us to control the exoskeleton and provide us biofeedback. The other technology we will be using in this study is the Spinex transcutaneous spinal cord stimulation. We have nodes attached to the T11 and L1 vertebrae, and these nodes provide dual amplitude frequencies. What's unique about this device is the 10 kilohertz carrier frequency that suppresses discomfort at the location of stimulation, which allows us to reach higher levels of stimulation. So we wanted to see if a single use of rehab technologies would impact step length for children with cerebral palsy. So participants came in for 4 randomized training sessions, one with them just walking, one with spinal stim only, one with the exoskeleton only, and one with a combination of the exoskeleton and the spinal stimulation. And we hypothesized that a resistive ankle exoskeleton will reduce step length in the short term because it's more challenging, while spinal stimulation will boost coordination of muscle activity to increase step length. So our methods, we had 2 participants for the study. Participant 7 is 6 years old and is diplegic, meaning that they had symmetrical paralysis, but in this case, their right side was slightly more affected. And participant 10 is 10 years old and right side hemiplegic, meaning that they had paralysis on the right side of their body. During each session, they walked on a treadmill for 20 minutes with a different rehab technology or a combination of multiple. And while they were walking, we used reflective markers to capture their position in space. So these reflective markers were attached to the participant's toe and heel so we could observe their position within the lab. We used Qualysys Track Manager to absorb and organize this data in order to create a matrix of position data that we could smoothly transfer over to MATLAB. So here you can see the reflective ankle positions and red force sensors that were created by the force plates inside the treadmill. And we, in MATLAB, we took the position data that was provided to us by QTM and created a code that would extract the left and right heel position out of a matrix of numbers. And then we had a code that computed those right and left heel positions into step length. And so our results, so on the graphs here, you can see the x-axis is going to represent the sessions that are occurring. So walking with no devices, exo only, and stimulation. And then we have the step length in meters on the side. And so after analysis, participant 7 had no increase in step length after a single use of rehab technology, except for in their left step length. And just to recall, their right side was more affected by paralysis. So their left step length is their least effective side. And here we saw a step length increase of approximately 7% compared to no devices. And then after analysis, or the slides are the same for participant 10, except for the inclusion of the exo and stim session. So after analysis, P10 had an increase in step length during every single assistive technology day compared to no devices. And they had the greatest increase in step length with the exoskeleton only. So they had an 11% increase in their right step length, which was their more affected side, and a 15% increase in their left step length, which was their least affected side. And then we originally hypothesized that the transcutaneous spinal cord stimulation, or spinal stim, was going to have the biggest impact on improved step length, but it turns out that the exoskeleton had the greatest impact in step length after one rehab usage. And we think that this may be because of biofeedback incentives that this method provides. So this involves setting a goal of how much someone is pushing off the ground with their foot, and then the participant trying to meet that goal. So creating a sort of game for them while they're using this rehab technology. And so ongoing work in this future studies are going to include larger population sizes and more time with each of the assistive technologies. And this is all with the hope that in the future, better understanding what mechanisms to incorporate to best help improve step length and assist children who have cerebral palsy. Thank you, and questions.
Questions? Yeah, in the back.
Great presentation. I'm curious on the rationale behind why to use the exo in resistance mode, and if you would have changed it to assistance, how that may have changed your results.
So we were using this as in resistive mode. The exoskeleton acts as a force that the participant has to overcome. And this is with the hope of them training that muscle group in their leg that's often weaker because of cerebral palsy. So in an assistive mode, the exoskeleton would actually help the participant lift their foot. And while this might be helpful for like walking in day-to-day life, we want to actually encourage strengthening of the muscle groups. And so we act in resistive mode when we're doing these sessions.
Okay. Thank you.
Thank you.
Is this open today? Is it open? Yeah.
If you're a summer camp student with Center for Technology, we're going to head out the back now. Thank you so much, Dr. Chudler and students, for letting us do this today.
Our next presenter is Nye Martin.
Good morning, everyone. This summer, I had the privilege of working under Dr. Cheperitz here at the University of Washington. And our research involved using spinal cord stimulation to improve walking in individuals with spinal cord injury and an exoskeleton. So our goal was to test participants using the exoskeleton assisted walking alone, and then comparing it to them using exoskeleton assisted walking paired with transcutaneous spinal cord stimulation. Now, why do we use transcutaneous spinal cord stimulation? Well, we use spinal cord stimulation because it sends an electrical current to the spinal cord, which facilitates a motor response. And this leads to more improved, continuous, smooth steps. The exoskeleton was very beneficial for this research. Some advantages is that it requires fewer therapists. Due to its structure, it is also safer. And studies have showed due to the active engagement of the participants, it improves their physical ability and quality of life. However, there are some disadvantages, one being there's a weight limit of 220 pounds. So for participants who don't fit this requirement, they're unable to participate. And it's also more restrictive. What do we use? How do we evaluate our data? So in this data, data in this presentation was taken from 2 participants. For our materials, we use the exoskeleton, this exoskeleton machine, and we also use the stimulating electrodes. With each participant, we did the assisted walking only first to determine the appropriate mode for them, and also to find we trained with stimulation to find the optimal amplitude range. We repeated this twice with stimulation off and then with stimulation on, and we tested for significant changes. And in this top diagram here, this just shows the 2 different modes that we used. So we used the trajectory control mode and the trajectory free mode. In trajectory control mode, the motion of the exo is guided and used to determine a gait pattern. Compared to trajectory free, the motion of the exo is determined by the participant. And so this mode is only for participants who can initiate a step. So for our study, one of the requirements is that you must be at least 1 year post-injury. So here are the results from participant 1. Who used the trajectory control mode. They were motor incomplete and had an injury at T10. And so these results show that as stimulation increased, their needed assistance from the exoskeleton decreased, which is good because this just shows that the spinal cord stimulation is resulting in more efficient movement, and so they need less help from the exo machine. For participant 2, they used the trajectory free mode, and they were also motor incomplete and had an injury at T12. So in this first graph, you see that we did the 10-meter walk test. So they walked at their normal pace and then at a faster pace, and both with stimulation off and stimulation on. And you can see significant changes with a p-value of less than 0.01, that with stimulation on, they were able to walk faster in both instances. In this graph here, we did a 2-minute walk test. So we just determined how far they could walk within 2 minutes. And once again, you see significant changes with stimulation on compared to stimulation off. They walked further. During the 2-minute walk test, we also looked at gait kinematics, and we witnessed these changes. So you can see during stimulation, they swung faster and they took longer steps compared to just the assisted walking alone. So after analyzing all the data, we can see that the addition of transcutaneous spinal cord stimulation improved exoskeleton assisted gait performance and coordination compared to just the exo assisted walking alone. With these results, we plan to conduct a follow-up study to investigate the long-term benefits of this combined approach, and which could also possibly restore movement, improving the mobility and quality of life for individuals with spinal cord injury. So here's the team that worked on the project, and I would just like to say a special thank you to Dr. Chet Moritz for allowing me to work in his lab, Rich Henderson for mentoring me, also Dr. Eric Chudler for putting together a great program, the National Science Foundation, Center for Neurotechnology, the University of Washington, and Florida International University. And also thank you for your attention. Any questions?
It's really promising sort of results. I'm wondering, in your last study mentioned longer term effects. One of the beautiful things about the work in the first lab is the plasticity that the stimulation causes. So there's a hypothesis that coupling that with the exoskeleton would maybe speed up recovery as opposed to doing stimulation alone. Do you have any thoughts on combined technology and what the longer term effects would be, where you no longer need either stimulation or the exoskeleton?
Yeah, so for this study, it was just a pilot study, so it was shorter, but we did still see, as you can see, some really great results. So in the future, yes, still pairing the exoskeleton assisted walking with the spinal cord stimulation. I'm sure like long-term you'd see like, even greater results than we did now.
Thank you. Our next presenter is Penelope.
Well, we're going to continue the trend of talking about transcutaneous spinal cord stimulation and spinal cord injury. But instead of focusing on lower body and walking movements, we're going to be focusing more on upper motor function. So what is spinal cord injury? I'm sure that none of you woke up today thinking, I might get in an accident and get a spinal cord injury and then be paralyzed. But in fact, any one of us can at any time and any age. In fact, the World Health Organization says about 1/4 to 500,000 people suffer from a spinal cord injury every single year. So we're going to quickly talk about a quick anatomy lesson on the spine. You see the spine? Oops, that's the wrong button. Sorry, there it is. Okay. You see the spine is in the cervical, the thoracic, the lumbar, and sacral sections. Notice that the higher up on the spinal cord the damage is, the more parts of the body are affected. So we are going to be just focusing on this cervical segment. But why is this even necessary? Aren't there already treatments available that help people with spinal cord injury? Well, yes, there are. For instance, physical therapy has been shown to actually restore function in individuals after spinal cord injury. And there's other interventions that helped ones to adapt to their environment. For instance, wheelchairs help people to get around. However, it's really important to know that just because you have a wheelchair doesn't mean that you get your function back and you get to walk again. And we're using this button now. And we're using this button now. Sorry. Technical errors. There we go. So what happens when we stimulate the spinal cord? Well, we see that we actually induce growth and reorganization of neural pathways. This has been seen to actually restore function in individuals. And specifically, as we've heard about in some of the other talks, transcutaneous spinal cord stimulation is the stimulation that we're able to deliver on the skin above the spinal cord. So the question that we're asking here is, since we know that physical therapy helps to restore function, and we know stimulation helps us to restore function, what if we paired the two together? Is transcutaneous stimulation with physical therapy more effective than just physical therapy alone? So like I said, we've talked about transcutaneous stimulation already, but one way to think of it is to think about this stimulation in two different waves. One, I'm going to call the carrier wave. This is a way in order to provide the stimulation in a non-painful way. And then there is the therapeutic stimulation. Take note that the participants in this study, we vary the frequency of this therapeutic stimulation in order to optimize the results for each of them. And also we gave a continuous stimulation. It wasn't pulsated or some sort of pattern. It was just a continuous stimulation when we were using that technique. Let's see. So thankfully, there was already a lot of clinical measurements out there to help us to determine how much function you have when you have a spinal cord injury. And these are the ones that we were able to use within this study. The GRASS test, I want you to take note of, specifically, it quantifies hand impairment. And then we have other studies like dermatonal sensation and muscle strength. This is going to be like, can the participant feel a pinprick on different parts of their body, or how strong are they on their right versus their left? We test their grip and their pinch strength, how much pain that they are feeling. We also take a spasm score. Most people who suffer from a spinal cord injury typically have spasticity issues. And we also find out how much independence do they have in their life. Are they able to feed themselves? Are they able to dress themselves? And then even though we have all of these measurements, we really don't know what kind of life do they live. So we take a quality of life measurements. Every single participant is going to go through the same timeline. First, there's going to be 4 weeks where we take baseline measurements with those measurements we just discussed. And then we're going to have an intervention. For instance, we might just do the physical therapy alone. And then we'll have a washout period. And then we'll do intervention again. And let's say it's stimulation plus physical therapy. We randomize both of these interventions just in order to get rid of any confounding variables. And then we wait 12 weeks and we do a follow-up to see if the physical therapy and the stimulation plus physical therapy had any long-lasting results. And our preliminary, our first findings have been very, very positive. So we see that with both physical therapy and stimulation and physical therapy, there's no decline at all. We've only seen improvements. In the blue here, you can see the average improvement after physical therapy is about 10% improvement. And then in the orange, we can see that after physical therapy plus stimulation, we noticed about a 45% improvement. And then you can see each of the lines here is representative of each of the participants in the study. Unfortunately, we do not have findings for the 12-week follow-up yet until all the participants have completed this. So I'd like to say that transcutaneous stimulation with physical therapy does provide a superior restoration of function than just physical therapy alone. There's some things that we could work on. We had a very small sample size. We only had 4 participants, and we really need to see, we need more diversity within our study. We also don't know if transcutaneous stimulation is safe for all. Like, what if somebody has a heart issue? Is this going to be safe for them to use? But keep in mind the big picture. We would like to someday see spinal cord injury patients be able to get their function restored, not just adapt to their environments. And my references. And I'd like to thank every single person who was involved in this study. The Restorative Technology Laboratory was amazing, and all the participants and everyone in the Endure program for facilitating this presentation.
Questions for Penelope? Yes.
So I have a question about the stimulation. Is this in any closed loop kind of setting, or just stimulation one that will pass? Can you get a little bit deeper on how the stimulation works and whether it could be improved?
Yeah, there is a lot of future work as far as closed loop stimulation goes. Right now, for the scope of this study, I'd like to say that the stimulation, we didn't use any stimulation during baseline measurements. That was completely the measurements of the individual without that stimulation. And the stimulation really was only done during the sessions that we were in that block of time where we had physical therapy plus stimulation. And then we're seeing the long-term effects of that. Does that answer your question?
Yeah.
Okay.
Thank you. And our last presentation of the morning session is by Dominique Cunningham.
Morning. Good morning, I'm Dominique Cunningham. I had the pleasure of working in the Pearl Meter Lab this summer studying spinal cord injury, or SCI. To give a little context about SCI, nearly 18,000 individuals annually in the US alone are impacted by it. And unfortunately, SCI typically comes with a laundry list of symptoms, some of which you can see on the screen. Because of the prevalence and the symptoms of SCI, the lab is dutifully working on rehabilitation. This is being done in long Evans rats. They're a great model organism, and is through this work that the lab has noticed a sex-based difference between the animals. As seen in the figure here, all the figures you see, these individuals have, or these animals have received a unilateral spinal cord injury. The ones highlighted here are our female experimental animals. All of our experimental animals received a spinal stimulation based upon activity, which reinforced the connection in the brain and improved their motor recovery. You can see as the weeks went by, the females increased their reaching performance, and even after therapy ended, this sustained. Unfortunately, with males here, highlighted in blue, this did not happen. There are several theories that males have higher anxiety in comparison to females, which would explain this, and this is why I looked for alternative therapies for the males. I had two groups here. I had 4 participants in my experimental group and 3 participants in my control group. The main purpose was to test whether social and environmental enrichment increases motor recovery in male rats via social interaction and exercising their injured paw. Social interaction was a large idea we wanted to implement here because typically rats are isolated in lab settings, which is unfortunate because this isolation can prove to be a hindrance in their motor recovery. From there, we have our hypothesis of the combination of social and environmental enrichment will provide a synergistic effect regarding motor recovery. And here I have my experimental timeline, and I want to focus on this acclimation period here. Because of hierarchical or stress-related reasons, rats can become very aggressive when they're paired together, so we decided to do some acclimation to lower the likelihood of this. This was done by a dual cage that had drilled holes. The holes allowed the animals to smell each other and become familiar with each other, as well as the acrylic barrier between them. They could not hurt each other, but they could see each other. And after 2 successful sessions of this, we used the rat castle with the barrier, as you see on the right there. This was used for 2 weeks. And after 2 successful weeks of this, we allowed the animals to come in contact for a week. And after this contact, we assessed their motor recovery by using the cylinder rearing test. We placed an acrylic cylinder over the animals and they stayed there for 2 minutes. We looked at how willing they were to use their injured paw, their uninjured paw, and both paws when they were experimenting and exploring the area around them. We then used the forelimb use asymmetry formula, and here are the results of that. The first 4 animals you see are the experimental animals, and the last 3 are going to be the control animals. You see a trend here that all of the experimental animals were willing to use their injured paw to explore the area around them, which cannot also be said for the control animals. But unfortunately, one of the control animals performed very well and actually outperformed all the rest of the participants. Because of this outlying behavior and of the small sample size, it's not accurate to say that we have any statistically significant findings. And is also when I was looking at this, I wanted to look if anything else could have an impact on the rearing behavior. And I remembered not all of these animals were injured at the same time. So I looked at their injury date in comparison to the rearing behavior, and unfortunately no correlation was found. To explain a bit more, KN60, 46, 56 were all injured on the same day, and they all performed differently. Moving forward, we want to give more time, longer than a week, to allow a bigger difference between the experimental group and the control group. And in addition to this enrichment will likely be done in conjunction to electrical stimulation, which is commonly utilized now and hypothesized to also induce neuroplasticity. I would like to give a thank you to my PI, members of the lab, Samir Morjani, another PI that aided me in this time, Eric Chedler in the CNT RE program, as well as the NSF and the NIH for funding. Are there any questions?
Questions for Dominique?
I have to ask one more. That was awesome. Were they all male rats in your study?
All male rats.
All male rats. So then tell me, why do male rats experience more stress? We talked about in the introduction than the female rats. What more do you think about that?
We're actually still looking at that, and it's hard to apply it to humans as well. So we're really just kind of looking at these rats. Some scientists theorize that males are just a lot more anxious after their spinal cord injury, which obviously will hinder their motor recovery. But yeah, we're still wondering.
Have you looked at hormonal fluctuations at all?
Ooh, we haven't, but that would be a really good thing to look at.
Okay, thank you very much, Dominique. That's the conclusion of the oral presentations by the students. Now we have a 1-hour lunch break on your own. The posters, hopefully if the room is unlocked, will be over here. But before everyone goes, again, I just want to thank the funding agencies, but I really, really want to thank all of the labs and the mentors who have accepted students for these 3 programs. I know that with the UW financial transformation, some of you have experienced some hardships, not only the labs, but the students themselves. So I really appreciate, here I am apologizing for the entire University of Washington. But But I really appreciate all of the effort that the mentors have contributed and the time and the patience that they've shown to their students. So I think that all the mentors in the labs deserve a round of applause.
Because without Eric, none of this would be possible, not only the symposium, but the whole summer coordination, and he's working behind the scenes all the time. And we are so grateful to all of you.
So again, enjoy 1 hour, hopefully not too long in the sun, and we'll see everybody in the poster session. If you're curious about when people are doing posters, these little QR tags at the bottom of the program tells you when the different students will be presenting.
Yeah, especially